the whole event is being streamed internally into CNAG. So there are quite a lot of people from CNAG who are sitting in the building somewhere else, and they will be seeing the presentations live, and they will come and join for coffee breaks and lunch breaks and so on. In addition, we have asked speakers whether they would accept to us to put the presentations on our YouTube channel. So if the speakers accepted for us to do that, then the presentations will go up on the YouTube channel, and people can then watch them once these presentations are up. So it's not that, so anyone can see it. It's not that we're trying to hide something. It's just we try to make it accessible. And unfortunately, the park doesn't have a bigger auditorium than this one here. So we're in the biggest auditorium when we go there. This event would not be possible without the support of the of our sponsors. We have had very many sponsors today. They are also in the room. They will give presentations that are slotted into the thing. The providers that sponsor us, they are, for us as CNAG, extremely important because they give us the technologies that allow us to execute projects that our users are interested in doing. So we try to put up a diverse platform of methods and expertise. We try to wrap around that all of the best practices how to do something, and then we deliver this to users. I should say, I don't know whether I have it in a slide, I should say that CNAG at the moment handles about 1,000 projects per year. So, you know, keep that in mind. We're about 120, 130 people at CNAG, but we handle 10 projects per person who works at CNAG. So this is quite impressive, and this is because of good organization of the laboratory, the implication of the people who work at CNAG, who every day come in and they really do their best to make to produce really good data for the people we work together with. Okay. This is the program. We will go through this over the day. I won't spend too much time on it. I'd hoped that you actually had a copy of this in your thing, but I noticed you didn't. In any case, this will come up at every break. There will be the upcoming talks will be shown to you. Okay. Good. This is this. Okay. So, brief introduction to CNAG, just history. CNAG has been around since 2010. We started in this building here in the Parc Scientifique. We essentially started with like 1,700 square meters of space, concrete, nothing else, walls like this, and we put a center inside that is capable of providing collaboration to people who work with us for their genomics-oriented projects. What is very important is that is our mission, our mission is to carry out projects in genome analysis that will lead to significant improvement in people's health and quality of life. And we do that in collaboration locally, Catalonia, Spain, Europe, the whole world. We work with clinical sites, we work with academics, we also work with industry. So this is sort of the perimeter. And the way we want to do things, and this is very important, we always want to do it at the best possible quality. Quality is the one thing we don't want to sacrifice. For those of you who take the tour later on today, we have obviously equipment that we can do our work with. So we have the sequencing laboratory, or where the sequencers stand. We have a sample prep zone. We have a biorepository. We have a big computer. Those taking the tour will go and see different ones of these things later on. Just quickly to the structure of CNAG, we have two big units. We have the sequencing unit and the bioinformatics unit, one headed by Marta, the other one by Sergi. A lot of people underneath contributing to all of this. At the same time, we have a couple of teams and groups, 8 teams and groups that do different areas and do research, but also collaborate with outside people on projects. So, you know, if you have a project with an interest in spatial transcriptomics, we have a specialist who knows how to do that and who will talk to you about your project. If it's viable, we'll help you with the project, do the project, and then help you in all the way from essentially the design of a project all the way through to the delivery of a result. That is actually the case for every project that we do. So we want to have projects where we can contribute to the design and make sure that the right things are being done, and then go to the end where the paper is written or help in the analytics, or basically help all the way through the samples, the laboratory, the sequencing, and the interpretation of the results, and then even go as far as helping you with writing materials and methods that go into the protocol and help with the interpretation of results if that is requested. I said quality was important, and sort of to underline the qualities, we have our quality system. We have a quality manager who stands outside our process and makes sure that our quality system is running correctly. We currently run under ISO 9001 and have an accreditation ISO 17025, which is a very high level of accreditation. And recently we acquired ISO 27001 certification. This is for data security, very important in the world of GDPR and things like that. And you've noticed that you've all been given a sticker that came onto you. That is part of the 27001, because the security essentially does not let anyone into the building. So being let into the building means you've been vetted in a certain way. And then we have also a certified provider status with Nanopore or Dovetail, and so we have a couple of plans to move out into new ISOs, and we work with the CEN, which is the European part of ISO, in the design of new ISO standards. For example, these two documents on the back are for clinical NGS. So, as I said, we do about just over 1,000 projects per year. This is sort of the areas that they hit. Most of it is human genetics, disease. Rare disease is a big part. Cancer is a big part. Those are the bits that many of the invited speakers today will talk about. And then, as you can see, there are like 240 distinct users, and we process about 15,000 samples per year. Just briefly, we have 7 areas of activity that we sort of try to define. Personalized medicine with projects like Impact, where we have multiple different impact projects that are running, are obviously very important. We put things like the 1+Million Genomes, which is a European initiative to share genomic data, including the clinically associated data of the donors of the genomes. We try to sort of share this cross-border and work on topics like federated learning across country borders. Very complicated things to do. Rare diseases is a typical problem that you can work on with genomic technologies. So we do a lot of projects in rare diseases. Same thing, cancer, a disease of the genome. We do a lot of projects that touch cancer in one form or another. We have single cell, which is a very, very big part of what we do now. We have genome in action, which is a different word for sort of epigenetic type of analysis. We have ventured into biodiversity because we're not alone on this planet. There are things around us that impact on us. Some of them live inside us, some of them live close to us. So we look at projects in biodiversity, and then we have an interest, obviously, with the technologies and the experience that we have that come through the single cell work or come through the cancer projects, also to venture into immune-mediated diseases.
So thank you, Ivo, and thank you, CNAG, for this kind invitation to present our work, which I will talk to you today about what we call the NAGIN program, which is a project for developing and evaluating a new personalized breast cancer screening strategy in Navarra. So this project is about personalized prevention of cancer, based on the fact that 40% of the cancer are preventable, and the cost of preventing a cancer is 1/6 of the cost of treating it. And of course, improving the prevention will add years to life and life to years. So what we already know about personalized breast cancer risk is that we know very traditional risk factors which are related to lifestyle and past medical history, and also family history. We already know this. And in the last 10 years, we have been able to identify what we call the genomic architecture of breast cancer risk that is made of very rare high penetrance cancer genes like BRCA1 and BRCA2, which produce a very high relative risk of having breast cancer. Also, we have some more or less common moderate penetrance genes, and at the very end of the spectrum, we have a very common low allele penetrant risk alleles. We could have even 1,000 of these, with a very modest contribution to breast cancer predisposition, but bringing all together in scores, we could reach even on a score which is similar to that of the high penetrant cancer genes. So if we already know all this about breast cancer personalization, why continue to do breast cancer screening based only on age? That is the question. So the hypothesis behind this project is that we could do a risk assessment for the patient and also stratify for the patient, and then be able to make a risk-tailored intervention that is reducing the intensity of breast cancer screening in those people with low risk, and an intensive screening for those in a higher risk. Oh yeah, but there are some questions which emerge. Is this, and does this kind of efficacy? That means, can we really identify the personal risk of a person having breast cancer? So the second question is, okay, what is the effectiveness of and about cost and benefits of doing this intervention? And finally, is this efficient? That means, can we do it? Is it feasible? Is it equitative? And would our population would accept this easily? So these are the questions, and this marks our goals. And for doing this, we explore some patients and methods. So we decided to take 3 different clinical risk groups. The first one is about 100 patients coming from genetics referral based on a very strong family history or a very clear phenotype of high risk cancer. The second group are those people, those ladies in the women in a moderate risk, which were followed by the public health system with a very special group. And finally, we have about 400 population risk, which are patients already participating in some previous of our studies, but not with being selected because of breast cancer. So at the same time, we have about 10% of our patients of our cohort being diagnosed with breast cancer and 90% not having breast cancer. So in terms of the exploration of the genomic architecture of breast cancer risk, we studied the monogenic susceptibility with a virtual panel of 12 high-risk genes and about 20 moderate risk panels. We also explored the polygenic risk score for one of the previously published and more well accepted, which is the PRS 313. And also, having all this information, we wanted to know about what we call the secondary finding. That is the opportunistic screening of secondary findings recommended by the American College of Medical Genomics. And also we added a panel of 3 genes related to reproductive genes and from pharmacogenetics for the GKB1A and 1B level of evidence recommendation. So in order to estimate the personalized breast cancer risk, we used the software by CanRisk made by the Bodicea group, which incorporate the personal history and also lifestyle, medical health, and also family history, and some of the polygenic risk scores and genetic conditions. So here are some of the results that we have up to date. In terms of the efficacy, we explore how many of our patients did have a mutation in one of the high and moderate risk mutation, finding that in group 1, that is the patients coming from for very high risk, the rate of mutation in high penetrance risk was 10%. In the moderate risk, we still have 4% of the patients which are not followed according to a high-risk protocol that are in this group. So there are still a number of patients carrying mutation in the general population that we do not detect. And even in the general population group, we still have some mutations, 1% of people having high-risk mutations. So to see what is the clinical validity of polygenic risk score to evaluate our patient's situation, we came from PRS 313, but found that some of the alleles, some of the SNPs, were not well covered by whole genome sequencing, and some of the alleles frequency in our population were not discriminating into breast cancer patients or non-cancer patients. So at the end, we came up with a PRS, we call that 286, with these number of SNPs, and we tried to evaluate the clinical validity of this PRS, finding that yes, it could predict effectively which patient will develop breast cancer at the end, and even this score will be able to discriminate with the groups of the clinical risk population. So in order to evaluate the efficacy, as you can see, we consider the clinical entry risk against the output personalized risk. So as you can see, only 16 patients in the clinical entry group did have a mutation, and the actual patients in moderate risk having an personalized moderate risk is only 1/3, and in the general population, 2/3 of the population were in agreement, but 1/3 was not agreed. At the end, only 40% of the patients have a matching in between the entry clinical risk versus the output personalized risk, having into account that about 70% of the patients not matching will have an actual lower risk. That means would need to receive less mammograms that they are receiving at the moment, and 1/3 will have an actual higher risk. That means that they will need to receive additional screening. So with this in mind, we decided to evaluate the effectiveness. So we consider some key performance indicators, having into account that in a program where we have 200 women entering the programs for 2 yearly mammograms from 50 to 70, around 50% of the women 50 women will have breast cancer, but in fact, only one of them will have additional benefits of a screening. So they will survive because of being diagnosed with breast cancer screening, whereas about 12 of them will have no difference in between having the screening or not. They will have breast cancer, but there will not be a big difference for them, and even 2 of them will be diagnosed with breast cancer and treated for this, but this treatment will not be changing the management of the patient as the final outcome. This is about the overdiagnosis. But also, we have that about 40 of these patients will need additional tests, with about 4% of mammograms resulting in false positive. That is, of course, the number of mammograms determines the number of false positive and overdiagnosis. And in terms of benefits, we need to consider the breast cancer mortality reduction, and also some additional benefits because of incorporating the patient with high-risk mutation to a specific management, and also to consider what is the benefit of determining this secondary finding for this opportunistic screening. So for this, one of our studies decided to evaluate on model 1, which would be to invite women at the age of 30 to fill in a risk questionnaire. So this will stratify the population in 3 different groups. We decided to study this model because in this population risk, we only found 1% of higher risk mutation. That means that we could explore this patient only by studying the polygenic risk score, and this will be enough to evaluate the risk. For the moderate and higher risk patients, we could then consider to go through whole genome sequencing with evaluation of high risk genes and also polygenic risk score. With this, we will distribute the patient in 7 different stratus and then propose a personalized risk tailored intervention going from triennial mammograms 50 to 17 years to, for those at higher risk, with a hybrid model of annual mammograms 40 to 50, and then going through a biennial mammogram screening 50 to 70, and incorporating patients from high risk to a specific clinical circuit. So with this in mind, we consider what we are doing now, and we know that the mammograms cost about 50 euros per exploration. The false positive rate would be 4%, with a cost of false positive of 700 euros. Overdiagnosed will mean about 1% in our population, with a cost of 30,000 euros. And at the end, if we proceed to this model I told you, doing ChIP-RY and whole genome sequencing for high-risk population, at the end, so at the current whole genome sequencing, considering a cost of 200 euros per whole genome sequencing, the Model 1 screening program cost will be approximately the same of our current program. So, and then we decided to consider what if we do whole genome sequencing to all patients. We probably would get additional benefits for other situations. And then we found that probably with this program offering all women in the age of 30 whole genome sequencing, and then incorporated to this personalized screening model, so the cost of the program would be about 10% higher than the current program in place. But this is only about cost. And when we consider what will be the benefits, so we evaluated that probably with this program, it will be 1 breast cancer death averted every male patients entering the programs. And because of the incorporation of the high-risk patients to a specific management program, we considered that we could obtain 25% life years gain per male patients exploring the programs. So we have not been able to evaluate what is the value of this detection of these secondary findings recommended by the American College of Medical Genomics, but there are some publications out there that propose to screen all people at the age of 30 because of the potential benefits of preventing only colorectal and other cancers and also cardiovascular diseases. So, and what about efficiency? We wanted to explore this about the susceptibility in our population. So we designed a questionnaire with a 5-point Likert scale going from 1 to strongly disagree to 5 points for strongly agree. So we decided to evaluate 5 groups. 3 of them were patients participating in the projects. Another group which was invited was patients from Breast Cancer Association. And finally, we invited doctors and nurses from our college, medical college, to answer the questionnaire. So the key question that we forward to this population, and probably would be good for you to have this in your own mind, is what do you think about the idea that the initiation and frequency of mammograms are based on your personal risk? The second is, if you had a higher personal risk of breast cancer, will you be happy to take more mammograms than the actual program? And finally, if you had a lower personal risk, will you be happy to have less mammograms? So here are some of the results that we obtained. So these are quite complicated. These are the questions here. These are the groups. So and I have to say that most of the patients, most of the group were strongly agree with this concept. But sorry, it has moved. So for the question number 3, you have a number of lower acceptance risk. That makes us to think about, well, if the cost and benefits of the program are mainly based on reducing the number of mammograms, will the population accept this? So, but we observe here another difference, very interesting difference here, which is that in the patients group, that is number 4, so the acceptance rates across the 3 questions were lower than in the rest. And we wonder why. And we came to the conclusion that it is because patients from the project, they did have a pre-test genetic counseling session informing about all the benefits of whole genome sequencing and these kind of things. And we presume that doctors and nurses do have this knowledge in beforehand. So they decide to do some information sessions for patient association, and then evaluated again the acceptance, and found out that the acceptance in this group was similar to the rest of the groups. This brought us to the conclusion that educating the population increased the acceptance of personalized screening for breast cancer. So the conclusion we do have up to date is that personalized risk estimation based on genetic and clinical factors has efficacy and shows clinical validity to assess breast cancer risk in our population. So second, that Model 1 and Model 2, which incorporate whole genome sequencing to personal risk stratified breast cancer screening program, seem to be adequate approach with so promising cost-effectiveness KPI. Finally, that comprehensive public education improves acceptability, and this needs to be incorporated to any planned intervention. So there are some parts of the efficiency that needs to bring together in a table to design our next step and intervention. So we still do have many data to be explored, and well, yeah, so we'll continue to working on this in the next year. So this project has been funded by the Navarra government with the participation of the University Hospital of Navarra and the Public Health Institute of Navarra. And this is a part of what we call the NAGIM program, which has been in collaboration with CNAG since I remember that's 2016 when we started the first programs. Up to date, we have recruited about 300 patients in different situations, clinical situations, to incorporate whole genome sequencing to the projects, coming from rare diseases to pharmacogenetics, hypercholesterolemia, urgent decisions in pediatric units, and also to infertility and reproductive medicine. So thank you very much.
Thank you, Angel. Do we have questions? Yes, there's a question here in front.
Thank you very much. I have to say that in BHIO here in Barcelona, we have a couple of projects that is exact the same, so we can talk later. We have started the last year, and we have now receiving the first pilot study that we call from CNAG, so we can talk later. I have a question about the variant of non-significance in the high penetrant genes and moderate genes in deep intronic regions, for example, that this is the extra benefit that can offer the whole genome sequencing. So do you have any pipeline algorithms to analyze them? do you plan in the future address the deep intronic variants, for example, because there are other variants of unknown significance in the promoter in the 5 and 3 untranslated regions. So that is that we are also, this is also one of the questions of our project. If the whole genome sequencing can offer an extra yield of diagnosis that not using whole genome sequencing, we cannot see that potential pathogenic mutations.
Yep. So in these results we have been exploring the clinical actionable mutations, which are those clearly classified as pathogenic and likely pathogenic. Of course, there are a number of additional benefits in other mutations that we could explore. We can very easily do this because we already have the data. But the benefits of this explanation is that you could have that information, which is a kind of research landscape, but you already have some clear benefits in hand that you could just directly translate into clinical benefits. So yes, we could have a collaboration in exploring that a little bit further. Yeah.
Thank you. Any other questions? Good. If not, thanks ever so much, Angel. Can you hit that? Okay. So it's my pleasure to introduce the next speaker, which is Sukhvinder Niklin from Pacific Bioscience. So she's going to explain to us our new machine that we have standing here in our.
Make sure I can use this. Good morning, everyone. Thank you very much for the opportunity to come and speak to you. I'm thrilled to be back here in Spain. So I'm going to give you a high-level presentation of what's happening from a clinical perspective, so the theme that Ivo set at the start of this talk. Now, I'm here with my lovely colleagues Chimo and Jules, so if you have any questions or want to know more about the details of the studies, please do come and find us in the breaks. I'm going to level set with this statement, so here I take off my scientist hat and put on my parent's hat. Even with a short-read whole genome, less than 50% of rare disease patients receive a diagnosis. So if we think about what does that actually mean, half the families out there with a poorly baby or child or adult don't know why they're living that difficult life that they're living. And I think that's the prerequisite for most of the work that all of us are doing. The Revio system, it's fantastic, it's here. We actually launched this instrumentation in 2022, and this was really pivotal to change the environment. This meant that suddenly the hi-fi genome was more accessible from a cost perspective and a throughput perspective. We had an interesting discussion around innovation at the speakers' dinner last night, and I think one of the things that we, PacBio, pledge to do is continue to innovate. And one of the important points about this translation into diagnosis is to have the full workflow from end to end. I'm not going to go into the detail of all this, but we address all of these areas. And on the theme of innovation, continuing with this, you may have heard of our Spark chemistry, so this will be accessible. What this actually means in a nutshell is a lower input threshold. So this means sample types such as saliva are much more feasible, and even beyond that, you know, the possibility of using amniotic fluid, everything becomes more viable. The other sort of key level points here are looking at the methylation, so the 5MC, the 6MA, and also being able to do 2 20x genomes on a single smart cell. This is accessible to you here in CNAG. Again, on the theme of having the full workflow, you'll see here there is a suite of software tools that are available to you, and this is continuing to grow. I already see I really do need an update on this, on structural variants. There's a new version of the software called Sawfish, which is giving you greater accessibility to actually understanding what's going on. A lot of these address the key rare disease areas that are relevant for us. The benefits of long-read sequencing, I think, probably preaching to the converted, but if you look at the top row there, understanding structural variants, understanding phasing, this is key when it comes to rare disease diagnosis. Repeat expansions, fully understanding the size, the composition of the repeat expansion is critical when it comes to things like the myotonic dystrophy, canvas, etc. This is all accessible using Hi-Fi Genome. If you look at the bottom line of figures, this is all around integration of omics. Now, not enough time to talk about that topic here, but this is something relevant for many, many areas. When it comes to methylation, you can really understand more around what's happening, for example, in imprinting disorders. Chromatin architecture, I know there's a couple of folks who are actually using FiberSeq, the methodology and the 5-methyltransferase enzyme to actually unpick what does the chromatin structure look like, where are those open regions, where are those closed regions, and when you layer that with the genomics or with the transcriptomics, the final figure, can you really understand what's happening within rare disease? Don't worry, I'm not going to go through all these, but there is so much happening right now, so it is really an exciting time to be in genomics. Projects that are going on, I point out in the middle, the Hi-Fi Solves, I'll talk a little bit more about that. The Estonia Biobank, the project around population genomics. Now, population genomics is holding hands with the innovations in rare disease and the advances in rare disease, because they will actually provide information about those variants, whether or not there is sort of a probability that this is due to the cause of the rare disease. So first part, population genomics. I want to talk a little bit about Estonia. For those that know, Estonia, and I'm embarrassed to say I didn't know a huge amount about Estonia. It's a country in the Baltics, south of Finland. One thing that I will say that I've seen firsthand that they do incredibly well is this translation to their population, this understanding of the relevance of genomics for them, the man on the street. And that within the Estonia Biobank, 20% of the population have actually contributed. That's incredible. We haven't done that well in the UK for sure. And I think this is something that's really, really fantastic about the work that they're doing. When I met them, they were looking at thinking, so we have 212,000 participants. They had about 3,000, just under 3,000 short-read genomes, and they were looking at doing more 10,000 short-read genomes, a small percentage of long-read genomes, and deciding which way to go. They also were looking at the pharmacogenomics. So the lead professor here, Lily Milani, is a pharmacogenomics professor and could see that there was benefit in that. So the work that they were doing around targeted pharmacogenomics analysis soon looked into, well, what can we do with the 10,000 genomes? And it's not a secret, you can see it here. Following the head-to-head study, they decided to go with 10,000 PacBio Hi-Fi genomes. And the reasons behind that were clear. It was more around the phasing of the haplotypes, accurate genotype imputation, phasing of the single nucleotide variants, and also the pharmacogenomics piece, understanding things like CYP2D6. The other point, final point I want to make about Estonia is they are going to translate this back to the population. So they'll use those 10,000 genomes to impute across all 20,000 within the biobank, and they will report back findings on breast cancer, on pharmacogenomics, and actually change the clinical pathways there. Now, going back to the area of my passion, what can we do in clinical? I've said to a couple of the folks that I met, things are really moving on in the last year, 18 months. Here you can see a study that came out of Sweden, and for those that know, Anna Lindstrand Larsvik, they put out this in the Långvid Uppsala meeting a month or so ago, where they talked about a 5-year vision, a 5-year plan of incorporating Hi-Fi into their clinical pathways. So starting in year 1, so this is what they can do right now, looking at the repeat expansions, the area that comes up and again, and then following in next year with looking at the pseudogenes and those areas of really complete deep richness, and then following on with imprinting disorders. Now, as we move along that line, we're going to be innovating as well. So the idea is that year 3 to 5, that they will be able to incorporate more and more along the Hi-Fi genome as we have our innovations ourselves. In a similar vein, I'm going to go to the Netherlands now. So many of you will have heard Professor Vissers or Professor Huyshen talking around Hi-Fi and the role in clinical and one test to suit them all. They actually carried out what they referred to as the Revio 500 study fairly recently, and they've published on the first part of this. But the aim behind that, in a nutshell, is can we actually identify all the difficult variants that are there? So they looked at about 100 or so samples. And the second piece to this was, can we actually get a diagnostic uplift? Is this feasible with the hi-fi genome? More publications to follow, but do come and find me if you want to know more. But the question that they were asked more and more is, if it's so great, why are you not using it routinely in diagnostics for rare disease right now? There was a really simple answer. I gave these discussions happened in Uppsala and also in Denver at the ASHG. What they stated was they needed a clinical utility study, which they have now nearly completed. So this is a study of 1,000 samples, and what they were doing was to look at standard of care comparison with the hi-fi genome to see, can we actually identify the same as we do in standard of care? Can we identify more? Do we identify less? Now, for those 1,000 samples, they chose a period of 1,000 samples in 2022. These are representative of the kind of diagnosis that they see in a year of their labs. It also represents the diagnostic yield that they find in their lab and the different myriad of workflow approaches that they use. The other point I would say is that this, and it took me a little while to get my head around this, this is a retrospective study because obviously they're using older samples, but it's also prospective because these people that are analysing these data are blinded to the outcome of standard of care. And what they will be able to do from a health economics perspective is actually look to see, so patient 2 didn't have any further testing in 23 or 24, patient 3 has, patient 4 has, then they can make really accurate decisions around the actual health economics and are we making advantages to what we're doing currently in standard of care from a cost perspective. So I expect to see publications and announcements around this in the next month or so. As I said, exciting time to be here. Now this is a map that is growing all the time. So these are clinical installs for the Revio. So really showing the difference that it's made in terms of Revio really facilitating that acceleration for hi-fi in the clinic. It's a growing slide, and I expect to see more dots in Southern Europe in the next few months. And yeah, this is something that you'll see, that there will be more and more individuals that are using the Revio and using the technology, especially with Spark chemistry coming on board, to actually change things. I want to talk about Hi-Fi Solves Consortium. So some of you may have heard of this, some may not. Hi-Fi Solves is the PacBio Long Read Consortium. It's a global consortium. So you see here the 3 main areas. If you look at the bottom first, in the Americas, they're really looking at things like the repeat expansions and what is the utility for the hi-fi genome in repeat expansions. If you look here on the right-hand side in the Asia-Pacific region, being led by KK Women's and Children, they're looking at infertility, so subfertility, so couples that may have had repeated miscarriages, what is going on there, or where there's a situation with male infertility or female infertility. So watching this space with great interest, because there are a number of folks in the EMEA region that I look after who are also interested in hi-fi within that area. Now, going to the top region, and I'll just switch to the next slide. This is the current Hi-Fi Solves EMEA map, and these are the lead institutes that are involved within here. Now, they are actually really interested in looking at areas of complex homology, so pseudogenes, and what can they understand from using the Hi-Fi within that area. Again, this group is growing, and when I present this again, even in as short as a month and a half, there will be more members within here who are actually going to accelerate the pathway. The areas that they're focusing on are twofold. Now, you can see similarities with the Radboud project. So the first point, can hi-fi genomes replace all testing effectively? So again, the group have looked at 100 mutation-positive controls. So these are the super challenging samples that really will see, is it feasible? This has been out for publication, and they found that they could actually identify most of them. There were some caveats, but we can go through that data if you're interested. The second point, I think, going back to my very first statement, is even more important. How many more diagnoses can they look at? How many more diagnoses can they identify? And for this, they're looking at 300 samples of undiagnosed rare disease, carefully chosen. So with the 300 going to the second half, they're looking at unsolved trios and unsolved singletons. They'll also look at those areas where the clinicians, the clinical geneticists are saying, there's got to be something going on here. There is a family with repeated indications, but nothing in all our sampling is telling us what's going on. Or they're looking at somewhere where there are multiple generations of families with the same issue. As I said, the group is growing. There is planning for 2025 happening, and that could be centred around, the next body of work could be centred around, although this is all whole genome sequencing, potentially targeted with the health economics parameters and the challenges that many of the governments have, my own included. Maybe that's an approach initially. Also looking at other areas of interest, so for example, the fertility area. I've given you a bit of a whistle-stop tour and hopefully have made up some of that time that we've lost, but I'm going to finish with another strong statement. Enabling the promise of genomics to better human health. This is the mission at PacBio, and a lot of the work that we do is centred around this, and this continuation to innovate, I think, is going to be key to make sure, as a couple of the folks said to me this morning, I'd love to do hi-fi. Cost makes it prohibitive. With the spark chemistry, with all the other innovations that are down the line, I think that's going to become more and more viable and make it more and more possible for folks like the lead institutes in the Hi-Fi Solves Consortium to actually take it into clinical. Thank you very much for your time today and for listening to me.
Thank you very much. Does anyone have a question? Yes, there's someone.
I have always the same question when I hear about the benefits of long-read sequencing. And I am very aware that the benefit is quite high. But comparing between the two existing currently techniques for long-read sequencing, PacBio, and comparing to Nanopore, what are the main differences between the two, and why we choose one or one other?
Yes, and there are friends here from ONT that I met last night. So to give an answer, I think there is a clear, it's no secret, if you look at the indels, there is a clear advantage with the insertion deletions. But I do, the work that I refer to, the Estonia Biobank project, I have all the data there. So please do come and find me and we can go through the data together and I can run through. One leaving point for you is what they did do in the Estonia Biobank is they downsampled from a 30x genome to a 20x genome, and they found no loss in what they were doing. So I probably should have said they are doing 20x genomes. And that is a conversation that many are having now, even in the rare disease world. What is that coverage? And now people are realizing that not all genomes are the same, and that ability, that work will be published, so you'll be able to see that data very soon. I think they're finishing off that paper now, but come and find me and we can chat some more. Okay, thank you. Thank you.
Thanks ever so much. Good. Thank you very much. So the final speaker before the coffee break is Ellen Iles. So yeah, from Valdebron. So thanks ever so much for agreeing to come here. See if I can get your slides up. There we have them. Good. Good. Next 25 minutes.
Thank you.
2020.
So thank you for inviting me. First disclosure is that I'm medical oncologist, but I promise you won't find any Kaplan-Meier in the presentation. And I have a question. Medical oncologists in the room, apart from you, Bulat. Okay, nice. I'm alone because he's not an oncologist at the end. So why to talk about colorectal cancer? Colorectal cancer is one of that we call a big killer. is the third most common tumor worldwide, and it's the second in mortality. So we have a lot of colon cancer, and we have a lot of colon cancer that is increasing in patients younger than 50 years old. That's what we call young onset colorectal cancer. So it is an unmet medical need, and we really need help to redefine how we are doing research in colorectal cancer. I really think that we are really backwards compared to other tumor types. It's true that we have incorporated biomarkers to treat metastatic colorectal cancer. Particularly, we don't have anything in the early stage, in the adjuvant setting, where we could have a wider window of opportunity to improve the survival of our patients, but what we have is in the metastatic disease. So we have learned that we have a proportion of 5% of metastatic colorectal cancer that are hypermutants, so they have an MSI high status or deficient mismatched repair proteins. So in these patients, we have the choice of immunotherapy, just a 5%. Immunotherapy has changed the panorama of all the solid tumors, but we have just 5% of our patients that respond to the treatment. And no? Yes. Really? There's a delay with my screen. Okay, so we have several additional biomarkers that you already know, like is the case of BRAF. We have BRAF inhibitors for BRAF V600E mutations. We have as well KRAS inhibitors for G12C mutations. We have now targeted therapy for HER2 overexpressed or amplified metastatic colorectal cancer. And we also know that for those patients without any mutations in the MAP kinase pathway, what it means, KRAS, NRAS, and BRAF, anti-EGFR therapies in the frontline setting favor overall survival, improve overall survival, progression-free survival, but just for patients with a right-sided tumor. We really don't understand what happens with right-sided tumors. Probably there's something going on with microbiome, but we don't have a clear explanation about that. Some groups are working in the role that overexpression of ligands, amphiregulin and epiregulin may have in this patient population, but it's what we have. So the panorama is quite sad. But if we consider three main features of colorectal cancer, we can understand why we have this restriction to treat our patients with targeted therapy. First, the colorectal cancer is a very, very heterogeneous tumor. Second, the genetic complexity, and it's frequent to find coexistence with diverse mutations, like is the case of KRAS, PI3K, that we don't know what to do with PI3K mutations, or for example, MSI high BRAF mutations. So the situation in which we have coexistence of these mutations makes sometimes difficult to know which driver do we have to treat or do we have to target. And then biomarker selection and patient selection. This coexistence will change along the time. So when we are doing a biological pressure to the tumor, then we are guaranteeing clonal selection, and the illness that we are treating at the very beginning is very, very different from the illness that we are treating in the refractory disease. So in the refractory disease, maybe we are working with a genomic profile that we did at the very beginning, at the frontline setting, or even in the early stage, because it's the sample that we have, and we are treating the tumor in the same way. So we are improving with liquid biopsy, but sometimes we don't understand what the liquid biopsy is telling us. So that's why we need collaboration with other disciplines. This is fundamental, and we need to redefine the design of clinical trials. So, and this goes to the next step, that is the concept of precision oncology. So at the end, the concept of precision oncology, and this is the same for all the solid tumors, is precision medicine, including the right intervention, so an appropriate drug, then the right patient, patient selection, and the right time. And this is fundamental. So referring to the right intervention, as I mentioned before, colorectal cancer is very heterogeneous. We have several techniques that we are using in the daily care. So in the daily care, we have an NGS panel, is quite restrictive. Paid by our public health system, we have a panel of 20 genes For example, we don't have APC in these 20 genes, so the information that we have at the end of the day is KRAS and RAS-BRAF, and that's it, because we don't know what to do with other mutations. For example, P10, P53, SMAD4, we don't have anything to target this particular mutation. So maybe we could have some additional information if we have information about the mutant allele fraction, something like that, but there is a lack of knowledge in the treating physicians to understand what we have to do with this information. So maybe it's fancy to have the mutant allele fraction, but we don't have anything to do in the daily care. And then if we have the chance of a liquid biopsy, and this is not reimbursed, in colorectal cancer, we don't have anything reimbursed coming from liquid biopsy, neither for the rechallenge. You may have heard about the potential to do a liquid biopsy in the refractory setting and then see if we have emerging mutations and then consider to reintroduce other therapies that we have previously used in the frontline setting. We don't have anything in guidelines and we don't have anything reimbursed. And if we have the possibility to do that, we have that option at VHIO. At the end, we have to distinguish what we are going to do. Sometimes we really don't understand what to target. Or for example, something very, very simplistic. If I have an all RAS-BRAF wild-type patient that have an emergent mutation of KRAS and has a mutant allele fraction of 1%, what does it mean? We have not established worldwide which must be the cutoff to decide if we have to reintroduce a treatment or not. So really, we don't have a chance to guide our treatment based on liquid biopsy unless we have a lot of expertise, a lot of knowledge, and we share this information with molecular biologists. Then the right intervention also includes what I mentioned before, the concept of the dynamics of the disease. Here you can see an schema of a patient of our department. He was very famous, a famous singer. Maybe some of you know him. He was a young onset colorectal cancer patient. He had 49 years old at the time of the diagnosis. He had liver metastases at that time point. The CT scan showed just one liver met and the primary tumor, and that's why we offered surgery to this patient. And when the surgeon was doing the resection of the primary tumor and this potential unique liver lesion, he found out that really the patient had 12 different liver lesions. At that time point, surgeons have to take a decision. He called us in the operating room. The majority of the lesions were at the same place, so we decided to go for the surgery. That's discussable, but it's what we decided to do. And as you can see, in the evolution of the illness of the patient, he never recurred in the liver, but we found as well a unique peritoneal node at the very beginning, at the first surgery, and then later on, all the illness the patient had was peritoneal, and he had as well lymph node metastasis. And what you can see is that the profile of the mutations really is changing along all the illness. So he had a BRAF V600E at the very beginning with a high mutant allele fraction, but at the end, he didn't have it. Just 1% of BRAF was found in these different tumor biopsies that we were performing. So this is a proof of concept of the genetic complexity of the tumor and the clonal evolution of the tumors and the need that we have to understand what we are treating. This is just a graphic representation. And then the right time. So here what you can see is just an schema because liver disease is the most frequent location of metastasis in colorectal cancer. So if I had to bet, I would say that patients with liver metastasis of colorectal cancer are really the unmet medical need in the panorama of the metastatic disease. So in the early stage, what it has shown to be very promising is to identify those patients that after the surgery have a ctDNA positive status. So this warrants later on the recurrence of the disease and probably with liver metastasis. So we have studied a lot which are the factors that favor a metastatic niche in colorectal cancer, but we have not found anything that could be treatable. This study of the group of Eduard Valle is very nice. They had the capability to identify cells in the primary tumors that later on could warrant the recurrence of the illness. And what is more interesting, and these models were MSS murine models, is that when you have if you treat these models with immunotherapy, then you have the chance to kill these particular cells so that you could have less recurrence in the coming future. But the issue is how we are going to find these particular cells. This in the daily care is very, very difficult because we cannot do this test to all the patients that are going for a resection of a primary tumor. But the issue is that it seems that the sooner we are incorporating the appropriate treatment for the patient, the better for the patient. So we are moving from the metastatic disease to the adjuvant setting, but now we have a lot of research in the neoadjuvant setting, because at the end, if we know that a positive ctDNA is a warrant to a future recurrence, let's try to have patients with less ctDNA positivity, and you may think that's logical. So what I can tell you is that there is no clinical trial today with a primary objective to decrease the incidence of ctDNA positivity in the adjuvant setting. So that's why I mentioned that probably the translational research is doing better and faster than the clinical research. And when discussing about the right patient, I'm particularly referring to young patients. So there is an increase of young onset colorectal cancer, remember, below 50 years old. Remember as well, and we had before a nice presentation about how important is the screening of the population. So the screening of the population in colorectal cancer is a fecal test. We perform this screening from 50 to 69-year-old because colorectal cancer is a tumor that is linked to age. So age is a risk factor for developing colorectal cancer. So these patients below 50 years old, are out of this scope. So we really have a problem. And the issue is that the registries that we have, at least in Spain, have not demonstrated an increase of young onset colorectal cancer patients. Maybe this has changed the last 2 years, but in the United States, they have started to do the screening at 45 years old, and they are considering to do that at 40 years old, because we are not catching these particular patients. It's very well known that these patients used to present with metastatic disease in 70% of cases, and it has been described that it's very difficult to have a suspicion about the clinics of these patients. So they used to consult for 4 to 7 times before a doctor considers that patient may need a colonoscopy because that symptoms are related with cancer. So what we have seen in young onset colorectal cancer patients is that from a genomic point of view, they are exactly the same. We don't find anything else. They have regular colorectal cancers with an APC, a P53, and usually a KRAS. We don't have anything. These tumors do not have fancy molecular aberrations, no fusions, no translocations, anything. So probably the increase of colorectal cancer in young patients is quite related to exposome, could be related with microbiome, but at the end, probably what the change is an epigenetic change that we are not catching using what we are using in research now and treating patients in the daily care that are genomics. So we are losing a way of patient selection. And can we move forward? So this is a schema that I would say we are still using. So first, we treated patients based on histology, of course. Then we moved to identify a particular target, as I mentioned, KRAS, BRAF, MSI high patients, and then we test the drug in a clinical trial for these selected patients. Now this has changed to another profile that is incorporating a lot of more of information. So it's very ingenious to think that if I'm just blocking BRAF, I will treat all the illness. That's why with targeted therapy, and that's very different from immunotherapy, I may have responses, but probably these responses are not profound and will not persist along the time. So that's why we try to incorporate using AI tools to incorporate information about not only one single biomarker, but different genomic signatures, incorporating as well radiomics, as well as pathomics, then generating machine learning models, validate that in an external cohort, and then turn that into, for example, signatures. There are some groups that consider that in colorectal cancer are the ones that could be considered born to be good, and those ones that could be considered born to be bad. And if we can identify these different profiles, integrating all this information, we could just preclude which will be the evolution of the patient in the future. So now, the treatment of metastatic colorectal cancer is, if I have a prognosis factor, namely BRAF, I have to give the patient everything, and that's 3 different chemos at the same time, together with a biological. So to give more chemo to the patient, that's a step backwards. We have to change the paradigm in which we treat metastatic colorectal cancer. So more chemo is not better for the patients. So that's why it's important to develop new different strategies. If I know that the patient will do poor along the future, I will prioritize probably quality of life and give the patient maybe more indolent treatment. That's the change of the paradigm. To treat, this is the contrary as we used to treat colorectal cancer. So these are the strategies that we have now. Basically, randomized controlled trials is the type of research with whom medical oncologists feel safe, and probably are the ones that offer a highest level of evidence and then turns into a change in clinical practice. Then for particular questions that may require lots and lots of patients, for example, 10,000 patients for a particular clinical question, like the duration of the adjuvant treatment, combined analysis of several clinical trials that we are developing worldwide could be an option. But the most interesting idea, I would say, that are this redesign of clinical trials. For example, in prostate cancer, the Stampede project represented a paradigm change in this sense, in which we are building clinical trials with different arms, different treatment combinations, incorporating translational research, and analyzing at the same time the results and changing the clinical trial in a dynamic way. So, So it's mandatory to collaborate, not only with medical oncologists, surgeons, etc., not only physicians or clinical arena, but with translational researchers. I'm sure about that. For example, you can see an example. I'm not going to explain all these clinical trials, but the NGIC network is the network that includes all the scientific groups that are dedicated to the treatment and investigation of GI tumors. And as you can see, are very simplistic. So these are the key opinion leaders developing clinical trials. And at the end, we have just treatment for elderly population with KRAS, for example, HER2 amplified. Here we have ancillary studies with pre and post tumor biopsies, nice, but this is less than 4% of all the metastatic colorectal cancer. The story that I mentioned before, and I would say that this is the more sophisticated one that we have, with overexpression of ligands, and for example, other low-frequency tumors in which we are just testing chemo. So the panorama is quite disgusting. Here what you can see, it's a potential. It's combining different clinical trials that are ongoing, that require maybe thousands of patients to answer a question, or for example, for very, very selected population to analyze all together the studies. But at the end, this will mean a lot of time and money to develop these clinical trials. So I would bet for this type of studies, in which I think you as researchers would be involved, because from the arena of medical oncology, I really tell you that there is a lack of knowledge about this. So that's why to have meetings, conversations, and think how we can synergize using your and our knowledge and technologies could be an extra. So what you can find is first, what I mentioned before, we have to collect the data and develop digital tools to incorporate all this information that now we are losing, coming from radiomics and pathomics and all the information about genomics that we can generate, and then developing clinical trials that are dynamic and in which the knowledge that we are generating will change the type of treatment we are doing and maybe the selection of patients that we are doing. And before at the presentation of the screening program, there was it was mentioned how relevant is to incorporate patients, because maybe we are developing something that seems to have clinical utility, may have an impact even in terms of economics, but the patients may not have adherence to that. So it's fundamental when we are developing collaborative efforts or international efforts to include patient advocacy groups developing not only clinical research but also translational research. So pay attention to this, because this will be very important, and even for particular treatments, regulatory. So why to invest in colorectal cancer or GI tumor? So if we want to build a project like this, incorporating all genomics and new clinical trial designs, we have to consider that these are big projects that may have a budget of 10 million. These must be international, collaboratives, must answer an unmet medical need worldwide, for example, tumors that are very common and with a lack of efficacy therapeutics. An important question to answer, so we would have, for example, colorectal cancer, we would have why we are seeing an increase of colorectal cancer in young patients, and room for improvement. So we don't have anything target for this particular patient population, and are the ones that could be that could have wider, as I mentioned before, window of opportunity to improve in terms of survival. So this would be really an area to invest. In particular, in colorectal cancer, this would be the winner strategies to move to earlier lines of treatments, and that would be, from a point of view, the neoadjuvant treatment that today it does not exist except for rectal cancer, very ancient, or, for example, trying to find response of the majority of our patients to immunotherapy. Why we have to do research in immunotherapy? Because with immunotherapy, those patients that respond, this response is maintained a long time. These patients could be cured just with immunotherapy, and this is something that we see with MSI-high patients. 95% of patients with metastatic colorectal cancer will not respond to immunotherapy. And this has been linked to the role of liver metastases. Now we know that for this 95%, if they don't have liver metastases, it seems that they could have the chance to respond to immunotherapy. So we consider that could be that liver metastasis may be modulating the immune global behavior of all the metastatic colorectal cancer. And important to stress that we see all these problems in younger and younger population. That's why we have to invest on that. And to wrap up, I think trials like this is an investigator-initiated trial that we are developing at BHIO together with Holger Hein, and we decided to tackle those tumors that are considered immune ignorant. So we have endometrial cancer, we have colorectal cancer, and for these both histologies, we have two different cohorts, MSI-high and MSS cohorts, and we treat these patients with a treatment combination of immunotherapy, and the main objective of this study is to perform pre and post tumor biopsies, and although we didn't find an enrichment of response, that was clinically expected, because we know that for MSS population we don't have particular responses to the treatment. We saw that the infiltration of, in this case, TCD4 cells would offer us the possibility to consider additional treatment aims to this particular patient's population. And what we are doing now, we have amended the study, we have lots of tumor biopsies, and now we are recruiting just for those patients without liver metastases. So we will have the chance to have information about the pre and post tumor biopsies of the liver and the pre and post tumor biopsies of different metastatic locations different than the liver. So with this study, and to my knowledge, is the only one that aims to answer this particular question, we may have the explanation about why particular patients with liver mets do not respond to immunotherapy and maybe generate a treatment strategy for this particular patient population. Indeed, at the same time, we are assessing matched samples from primary tumors and liver metastases and doing the same analysis. So to end, my key message is that it's fundamental to speak one with the other. So we have to find the time to discuss about clinical and medical needs, different ways to answer these particular questions, and always thinking about patient-centered intervention. So if we spend a lot of time and money developing tools that may not be useful or feasible in the clinical arena, we will be spending our time and our money. So we have to talk to each other, and opportunities like this, like today, are fundamental. So thank you for your attention.
Do we have questions? God, everyone needs that coffee urgently. No, but
Everything clear.
This is totally clear. No, thank you very much for enlightening us on this. This is much appreciated. Okay, good. There's going to be a coffee break. It's scheduled for 30 minutes. I will at some point start making noise out there, and then you can start coming in. So I'll try to leave enough time for you to talk to each other, because this is always very important at meetings like this.
Dear colleagues, dear partners, thank you for this opportunity to present Erdera during this 13th CNAG Symposium on Genomic Research. I am Yanis Memouni, the Assistant Coordinator of the European Rare Disease Research Alliance, working at INSERM France. So before talking about Erdera, I just would like to remind that this Horizon Europe partnership stems from more than 20 years of efforts that covered all fields and that contributed to the development of the rare disease research ecosystem. This covers the development of high-level strategy and investment, the integration of patients' needs at the center of all activities, the reinforcement and building of relevant research infrastructure, and bridging healthcare and research efforts. These efforts were already encompassed by a previous program that was named the European Joint Programme on Rare Diseases, EJPRD. So EJPRD aimed at ensuring rapid translation of research results into clinical application and uptake in healthcare for the benefit of patients. This was done by expanding and sustaining rare disease research funding, creating virtual platform of data, tools, and resources, building and enhancing specific education and training activities for patients and researchers, and finally, organizing dedicated supporting services to accelerate research results into tangible outcomes. We trained more than 10,000 stakeholders, including patients, researchers, clinicians, and we invested 104 million in collaborative research projects, funding more than 200 projects, and developed resources, including an interactive innovation management toolbox and the rare disease virtual platform that connects various rare disease research resources like patient registries, biobanks, catalogs, knowledge bases, data deposition and analysis platforms. We also contributed to the development of standards and analysis workflow, to which CNAG was a key contributor, like for example the Beacon framework development and GPAP functionalities. However, there are still remaining needs. 95% of rare diseases are disregarded in terms of research or lack an effective therapeutic options. 50% of patients still do not have a confirmed molecular diagnosis, and on average, 4 years are needed to be diagnosed when the rare disease is known. 52% of carers, patients, and families report a severe, not only an impact, but a severe impact on their quality of life. So we continue our efforts in connecting the dots to improve the health and well-being of 30 million people living with a rare disease in Europe, aiming at reducing the diagnostic odyssey on average to 6 months after coming to medical attention. We want to contribute to the development of new effective therapies that are approved specifically for diseases without approved options, and to better understand the burden on rare disease on patients and their carers and societies, to improve the quality of life and influence policy decision making. So this is how we are aiming to reach these 3 impacts through this specific impact pathway that starts with the operational objectives at the bottom, where resources and means are exploited to produce short-term and medium-term impacts in the middle, which ultimately will contribute to achieving these three aims. This is Erdera that is going to contribute to these aims, that gathers now more than 180 organizations. We grow since the proposal submissions, and we are having together all the national research funders, research performing organizations that are the most predominant stakeholder in the typology of organizations. We are having patient organizations directly involved not only in the implementation of research, but also in the strategy and its evaluation, and in every bit that we are going to explore together. We have, of course, university hospital, not-for-profit and public administrations, and three research infrastructure for clinical research like ECRIN, translational research, EATRIS, and for biobanking, BBMRI ERIC. In addition to that, this public-public partnership gathers as well private for-profit entities, including the biggest pharmaceutical industries and SMEs like biotechs and IT and data management SMEs that are contributing together to all these goals. We gather 37 countries, all the EU member states, the associated countries, and non-EU countries. When we look at the different type of activities of Erdera, they are represented in this house, where we have the coordination and strategy that are guiding two main blocks of activities. The first activities are the rare disease research funding, where we are going to continue to fund joint transnational calls for basic, preclinical, translational, and social science and humanities, but also we are going to launch clinical trial call in order to fund clinical trials by 2026. We will continue to have this specific funding for small networking events. Well, small, it's 30K per event, in order to allow you to organize events, workshops, meeting, in order to expand your network, to gather consortia to apply to the different calls, and to exchange on knowledge that is gained. This is the part on the funding. When we look at the other block, it's what we call the in-house research activities. This is research that we are going to perform through our clinical research network that is divided in three modules. The first module is about the diagnostic research that we are going to dig a little bit more into in the next slides. The second module is about the clinical trial readiness and what we call the outcome research. We have a part on the real-world data where we are going to exploit the electronic health records to have their automatic extraction. Then we are going to build and improve the building of different cohorts and rare disease registries and connect them between themselves and to national databases and national registries in order to have them regulatory upgraded to be used for post-authorization safety studies, post-authorization efficacy studies, but also with the aim to have a regulatory context, a context of use, to be used effectively in clinical trials. We are going also to develop a clinical trial, virtual clinical trial simulation platform, where drug development tools are going to be developed in order to guide the acceleration of clinical trials. In addition to that, we have a part dedicated to the patient centricity through the patient-centered outcome measures, where we are going to continue to maintain and develop all the patient-centered outcome measure databases to see if the existing tools can match group or some rare diseases, develop new ones through innovative methods, including connected devices that are directly linked to patient registries, but also build a framework to have the medico-economic evaluation of the burden of rare diseases in order to influence the policy decision-making when it comes to reimbursing a specific treatment, a specific newborn screening, or the implementation of diagnostic strategy within healthcare systems. Then the third module is about the innovative therapies. And there we have two parts. We have a part on the advanced therapeutic medicinal products. We are aiming mainly on gene editing, where there will be the ranking of the diseases and group of rare diseases that may benefit of the ATMPs. Then this group of diseases or rare diseases will be matched to technological platforms that are dedicated to gene editing and vectors that I'm going to present afterwards, in order at the end to launch a proof of concept studies where we are going to test these strategies of standardized technological platform and ranked rare diseases to have a preclinical validation, and ideally launch clinical trials to develop ATMPs. This is the part where most of our pharmaceutical industry partners are involved. In parallel, we are going to have an academic-focused approach where we are following the N-of-U approaches, where patients with nanomutations are identified, for example, through the activities of the diagnostic research. Then there will be antisense oligonucleotide development through platforms that are managed by academic partners for the development of the preclinical package before having the drug directly being delivered at the center of care of the patient in order to and the follow-up of the outcome of these therapies. These two blocks of activities, the research funded one and the in-kind in-house research ones, are supported by transversal services. We have services that are linked to data when it comes to connecting the different resources and make all the data that is being generated findable, accessible, interoperable, and reusable through the virtual platform, but also through different tools and methods that are developed in order to make the data and the processes machine readable, including the analysis processes. We are having a part dedicated to data sharing and analysis itself that I'm going to detail because it is much linked with the topic of this consortium. And we are exploiting also the different ontologies and semantic models that are available to enhance knowledge, use prior knowledge in order to have better analysis and better insights into research. We are providing also expertise services where some projects are guided in order to avoid what we call the valley of death, so that when the preclinical project is aiming to be translated into clinical development, they are being accompanied in order to ensure that all the regulatory guidelines are applied, all the different study, careful planning is there. We are providing regulatory support as well, and the support for the implementation of innovative methodologies for preclinical research and clinical research. The training hub is addressing knowledge gap by providing some trainings that are addressing some pre-identified gaps, and we are going to develop a little bit more about that in the next slides. And then the accelerator hub, in addition to the technological accelerator, we have a public-private accelerator where we are going to scout for research projects that are mature enough and have a promising probability for translation into clinical development to provide an effective therapeutic solution, whether be it a drug or a validated diagnostic device or validated biomarkers. These projects are being accompanied in order to find further funding to accompany them for the next step of development. And of course, we are not reinventing the wheel, and we cannot achieve our aims alone. So we are sitting on the international and national capacity alignment, first through the constitution of national mirror groups within each country, where different stakeholders from the rare disease community in each country, including the regulators, big research infrastructure, key opinion leaders, key researchers and clinicians and patients are sitting together in order to assess and communicate on the needs for rare disease research within the specific country, see how Erdera activities and results could address these needs, and the other way around, should there be any resources in terms of capacity, expertise, infrastructure that are available on the national level, align them with our activities for a better synergy. We are having a specific focus on underrepresented countries as well in order to accompany them and not leaving them behind so that they can benefit from Erdera's results, and the other way around. We could benefit from their flexibility, for example, and the global collaboration is ensured through many activities, including the International Rare Disease Research Consortium, which scientific secretariat lies within Erdera, but also links with other clinical research networks that have been identified and with whom we have established some links, and alignment with all the different research initiatives and missions and programs that are listed in our strategic research and innovation agenda. So there are immediate opportunities that are offered by Erdera. In addition to the yearly joint transnational calls for multinational research projects, our networking support scheme that finances events and workshops to share knowledge and build rare disease network, and the dedicated call for clinical trials, there are open opportunities to engage in in-house research that encompasses diagnostic and therapy development, on which I will focus in the next slides. But first, I would like to remind you that today, 10 December, we launched the 2025 Joint Transnational Call for Development and Validation Preclinical Therapy Studies for Rare Diseases using small molecules and biologicals, to which you can apply, and you can register to the webinar scheduled for the 17th of December that will provide general information on the call, explain the scientific goals, the administrative aspect, the support services, and the patient engagement strategies. Let's look at the in-house research activities that are closely related to genomics and their translation into healthcare provision. Improving rare disease diagnostic in Europe will be achieved through 5 targets: connecting rare disease experts across Europe and enable clinicians, scientists, and patients, including from underrepresented countries, to participate in the diagnostic research network; coordinate and consolidate the pan-European effort in diagnostic data availability and collection, including the standardization of data, its harmonization, following FAIR principles and quality control; then reanalysis of the data, so data reanalysis leveraging on existing knowledge and developing best practices to improve the variant interpretation across disease groups and European reference networks. This will allow the discovery, for example, of new gene disease associations, and expanding the reanalysis activities with innovative genomics and multi-omics approaches, while supporting complete genome analysis specifically for underrepresented countries. And last but not least, facilitate knowledge adoption for routine clinical practice and share best practice guidelines, particularly for innovative approaches that significantly reduce the diagnostic timelines. We are having in this diagnostic research module 3 work packages, the work package 6, 7, and 8 of ERDERA, that are aiming to achieve these targets. And you can see here how they are interlinked, building one on the other. Work package 6 will focus on data availability and quality, forming the foundation for the research activities in work package 7. This work package 7 emphasizes reanalysis of existing genomic data by developing new analytical methods through data analysis task force and providing interpretation of findings through thematic and collaborative data interpretation task force. Altogether, providing implementation guidelines on reanalysis strategy. The work package 8 extends the work package 7 activity by introducing complementary activities, including complete genome analysis and multi-omic approaches. So the ERDERA diagnostic framework is transitioning from a distributed approach to a federated approach over a 7-year period. This is done through 3 phases aimed at accelerating diagnosis. In the phase 1, we have the distributed approach, where phenotypic and genomic data are uploaded for a coordinated analysis and interpretation, providing feedback to patients. And this phase targets local healthcare providers with limited infrastructure or bioinformatics supports. The phase 2 is an enhanced distribution approach, where analytical tools and interpretation guidelines are provided for local data analysis and interpretation. Results are then shared with ERDERA for broader insights. This phase continues to support the healthcare providers that are lacking advanced resources while refining processes. And then the phase 3 is the federated approach, where data access is expanded to include other centers, enabling combined local and federated interpretation. Results are shared within ERDERA, promoting collaborative advancements, and this phase caters to national program and industry partners, ensuring scalability for broader adoption. So you can see that this evolution builds on prior examples like SolveRD for the distributed approach, and aligned with infrastructure initiatives such as 1+ million genome and the genomic data and infrastructure, GDI, for the federated one. Knowledge dissemination is key for ERDERA diagnostic component. The guidelines play a critical role for translating research into clinical practice. In this part, we are integrating international guidelines on genomic variant interpretation, tailoring them to clinical diagnostics, and training sessions are planned for healthcare professionals and researchers. The in vitro diagnostic regulation also frames our efforts, ensuring that innovation aligns with the clinical and regulatory standards. And additionally, knowledge bases and ontologies like Tritabolum, that is a gene and variant-centric database for rare disease treatment, and the medical action ontology will enable clinicians to link the genetic findings with actionable treatments, ensuring that diagnostic innovation impacts patient therapy as well. So ERDERA embodies a synergy between national and EU infrastructure. We are not working alone. So we are building on Solvardi. We will leverage on a large-scale research ecosystem to address undiagnosed rare disease cases, collectively advancing diagnostics and patient outcomes. The interconnection of international healthcare system, including diagnostic rare disease centers, centers of expertise, and European reference networks, with the clinical research network of Erdera, will drive this innovation and new diagnostic opportunities. The link with the genomic data infrastructure will foster our federation efforts, as said previously, through providing secure access to genomic data for improved research, policymaking, and healthcare delivery across Europe. And the collaboration at the national level through the national mirror groups will be further fostered through the links with the Beyond 1 Million Genome. Then, when we zoom on the data services hub, and specifically on the data sharing analysis services, this is the work package 15 that is establishing a robust data sharing and analysis infrastructure. The key objectives include deploying a scalable analysis platform compatible with cloud services, developing high-performance computing protocols for large data sets, and enabling federated analysis use cases, and the focus starts with diagnostic research. Looking further at Erdera's activities contributing to translating genomic into healthcare provision, we have the technology accelerator. So there will be technical developments, standardization, regulatory qualification of several key technology areas, including the AAV vector production, therapeutic RNAs, mRNA-based nanoparticles, gene editing approaches, and immune-mediated therapy responses. And training are key for Erdera and are part of our sustainability strategy. So Erdera's training program empowers young researchers and patients across all levels of rare disease research, beginner, specialized, and advanced, to focus on areas like provide training focusing on areas like pediatric rights, ethics, orphan drug accessibility, and genetic disease specificities. It's not only about the young researchers and the patients, but we are also having targeted multi-stakeholder training, addressing genomic data management for reanalysis, innovative omics exploitation, and advanced clinical trial methodologies, ensuring that all these different stakeholders are equipped to accelerate diagnosis and clinical research for rare diseases. And finally, we aim to develop a European curriculum that establishes a robust academic program in the form of an EU recognized diploma, like an EU master, where we are starting to integrate transnational learning and ensuring sustainability through strategic partnerships across academia, starting with all the universities that are involved. Beyond Erdera, so we are not evolving alone. We have established already many links with the several initiatives, like the personalized medicine partnership when it comes to funding and data services hub, the genomic data infrastructure and Jardin for the clinical research network, and the data services hub as well. In the acceleration hub, strong links with the innovative health initiatives are already being established, and we are establishing for the education and training links with the EIT Health. We remain available should you have any question through this email address, erdera@egprairediseases.org. So please do not hesitate to send us any email, any question through this email, and we will ensure that we are providing a comprehensive response to you. Thank you so much, and see you through other online events or in writing.
That was a very animated presentation. So really, really very nice. I have to tell him the moment I see him next time in person. Good, very, very well. Good. It's my great pleasure to invite the next speaker, who is Leslie Matalonga, who is from CNAG. So 25 minutes.
Yeah. So thanks, Ivo, and I'm very happy to be here today to give to explain some of the results that we have here at CNAG, working on diagnostic research towards clinical applications. So as Ivo was mentioning this morning, at CNAG we have different strategic areas. And today I will be mostly focusing on the first two, the personalized medicine and rare diseases. And basically the work I will be explaining after really involve many, if not most, of people working here at CNAG, from the scientific and operations, but also the sequencing unit, which is led by Martha Good, and the bioinformatics unit that is led by Sergi Beltran. And all of these with this layer of quality and certification accreditations that we have implemented here. So also to have an overview a bit on the amount of the activity that this represents, we have pointed here. It does not change as okay. So we have almost 40% of projects that are related to these disease genomics, which can include the personalized medicine and rare diseases. And this means that around 5,000 to 7,000 samples are processed towards these two areas. And when we speak about personalized medicine, what we meant is to try to analyze the genomic background of individuals to understand which treatment will be more suitable for that person. And not only this, but it's also to ensure that we won't be giving a treatment that won't work, or even worse, that will have negative effects. And when we speak about rare diseases, we speak about these 6,000 to 8,000 disorders that affect less than 1 into 2,000 individuals, and that have, in 70% of the cases, genetic origin. And also it's important to take into consideration that 70% of them start or affect childhood. And it is in this context that also at CNAG we developed the RDConnect Genome Phenome Analysis platform, and this is work that is led by Sergi Beltran and David Epicia, where we develop a platform that enables collation, sharing, analysis, and interpretation of rare disease data. And basically, what it does is to take and integrate clinical information and genetic data for downstream analysis and interpretation. And this platform is already used across Europe, and there are more than 30,000 genomic data sets that are available there. And also based on the different needs that we have had at CNAG, on different projects of programs we are involved, we have developed different instances of this platform that are used in several of the pilots that I will be explaining later. And also importantly, and Yanis mentioned it earlier, we are also working towards the IVDR compliance. And for those of you that are not familiar with IVDR, so it's a kind of new European regulation for products, in vitro products that are used into diagnostic. And this regulation is quite more complex than the previous one, and it really means that we need to start developing our products, methodologies, thinking on these regulatory requirements so that they can be applied later on into clinics. Otherwise, doing it retrospectively or trying to use a research tool into a diagnostic setting, if this has not been taken into account, will be impossible for hospitals to manage. And to ensure this validation or this quality, it's also important to, while we are developing or creating these platforms, to go through external quality assessments. And these encompass, for example, participating in quality assessment like GenQA that will validate the analysis and interpretation of the data, interlaboratory comparisons, where CNAG is also highly involved, or the use of gold standards that are available, data sets that are available for the community to benchmark analysis and tools. And this will ensure reliability, accuracy, and then help for the clinical implementation. So now what I would like to show you are the different pilots that we have somehow participated at CNAG regarding first the reanalysis of unsolved cases, the application of whole genome sequencing for rapid clinical management, the use of genomics for treatment suitability, and finally the use of genomics for early detection, mainly regarding genomic newborn screening. So the first one is on reanalysis of unsolved cases, and it has been mentioned before. It's important to keep in mind that almost 50% of rare disease patients will remain without a diagnostic after going to the whole routine hospital workflow. And this is why across Europe, different groups and countries decided to join efforts under the umbrella of the Solvardi project. And in this project, we did reanalyze more than 90,000 data sets and also sequence new evidences for trying to identify the genetic causes behind these unsolved or undiagnosed patients. And there were also part of the work on validating these possible novel genes. And this is really meant to bring equity on diagnosis across Europe. So a patient, independently of where he's from, should be able to access this type of a complete reanalysis. And the CNAG participated at different levels. So in the infrastructure, providing the RDConnect GPAP, the platform I mentioned earlier, and also at the analysis level, where Sergi led our package on this, and where we did reanalysis on exome sequencing, also genome sequencing and RNA-seq. And also we participated by doing the sequencing, and this is led by Marta Gut and Esteve. So here it's a bit of the workflow that we use, but I won't enter into details here, but basically I would like to give the message that what we did to be able to reanalyze more than 6,000 cases was to develop methodologies, systematic methodologies, that would enable this reanalysis possible, to take into the genomic data set, the phenotypic data, and also pedigree information, and to deliver these comprehensive analysis suites, meaning these systematic analysis approaches, and to also include a two-level expert review. And this means that we really work in tight collaboration between the analysts from the different research centers, such as CNAG, together with the molecular and clinical geneticists from European reference networks. And this is an example of this systematic reanalysis, and this one is on SMV and indels. And for example, here what we did was to develop a programmatic access of the RDConnect GPAP. So this way we could reanalyze in batch many cases and propose candidate variants, or pinpoint which variants could be interesting or likely pathogenic, so that the interpretation task forces, the clinical geneticists and clinicians, could go back to the cases and do the interpretation on these variants and decide if those were of interest, or confirming a diagnostic, or were rejected. And by doing this type of analytical and systematic approaches, this was done also for other type of variants, it enables us, after analyzing these 6,000 cases, to identify a diagnosis in 800 previously negative cases after having an exome or even a genome. And most importantly, the amount of time that was invested by experts, the individual expert time, was of around less than 5 minutes per variant. And this is quite relevant because until now it's very difficult to have reanalysis implemented into clinical practice due to the overload of the workload that they have. And this way we are closer to follow the ACMG recommendations on doing reanalysis, reevaluate cases every 2 years. And I also mentioned that within Solvardi we have done new sequencing to try to have more evidences and identify this genetic diagnosis. And here is just to show you that we worked with whole genome sequencing. We also did pilots on long-read whole genome sequencing, and this is part of the work that you've seen before from PacBio, and on RNA-seq. And for example, in this first one, which is a work that we did in collaboration with the Hospital Sant Joan de Déu, they prepared a methodology to do a trio whole genome sequencing together with RNA-seq, and they managed to solve an additional 40% of the cases. In the example on long-read whole genome, they managed to identify a diagnostic in 13 additional percentage of cases, And for RNA-seq, which work is led by Jan Esteve, they managed to identify 7% of extra diagnoses, and they also have many other candidate variants that are currently under study. And this is also to illustrate that this type of projects are already developing methodologies and systematic analysis to after once these technologies can be available in clinics, we will be ready to give or to propose protocols to analyze and interpret this data. And all this work within Solvardi gave rise to many scientific publications, but most importantly, it gave rise to additional knowledge. So we identify new disease-causing genes that are now possible to screen into clinical practice, and also we put all the information on interpretation of the data in ClinVar, which is an international database, so that it's really available to the whole scientific community when they are currently interpreting the data, also at the hospital level. And as Yanis was mentioning before, now within Herdera, what we want to do is really to go one step beyond, so not doing this centralized approach and having people sending the data, but really bring the expertise and the infrastructure at the national level, so that all countries and centers can have the opportunity to give these this type of pipelines and technologies for the rare disease patients. Okay, and the next pilot I would like to present is on whole genome sequencing for rapid clinical management, and this is work that we've done in collaboration with Navarra Biomed, that are leading these NAGEN programs, and Angel before presented NAGEN mix. So this one is NAGEN Pediatrics, and the objective here is to analyze the complete genomes of pediatric patients for rapid clinical management. So basically, this is the workflow of the work we did. So at day one, they recruited the patients with a multidisciplinary evaluation. Then they did genetic counseling and deep phenotyping, and this is a very key relevant step because they were doing deep phenotyping using the HPO ontology, meaning that the symptoms of the patient are computer readable, and we could use it then in the GPAP system to analyze, prioritize, and interpret the data. They extract the sample, and then at CNAG we perform the genome sequencing, the data processing with the CNAG pipeline, identify SNV indels, CMVs, other structural variants, STRs, and round of homozygosity. And we made all these results available in a dedicated instance of the GPAP. Then we did variant prediction and interpretation, and this was done in collaboration between CNAG and Navarra Biomed in parallel. And then when they had interesting findings, these variants were validated by Sanger, and they issue a report. Most importantly, these results were then communicated to the families, and these under the genetic counseling within the Servicio Navarro de Salud. And apart from this, during these years, from the different projects under these NAGEN programs, we have been transferring this knowledge to NACERTIC, which is the supercomputing center in Navarra, and indeed Ibai is here today in representation, so that now all this part can be done there. And these are the overall results of this project. So in total, there were enrolled 259 patients, and most of them were analyzed in trios. So this is where we have sequenced the genome of 751 individuals, and the average age of recruitment was 3.8 years, and the turnaround time to deliver results of 18 days. And regarding the interpretation, in total 1,628 variants were interpreted, and all this knowledge is now available within the system. And indeed, when we did the interpretation, we were doing it in a collaborative way, even if some of them were here or in Navarra. 259 reports were issued, and as overall results for the intensive care unit cohort, 39 patients got a diagnostic. And most importantly, from those 39 patients, more than half of them, this information did have an impact on how the clinical management was done after all, meaning that without having this information, they would have done something different as was better for that patient. So basically, as conclusions here, whole genome sequencing is a useful, rapid technique with a good diagnostic rate and clinical impact in hospitalized children with, of course, suspected genetic diseases. And this study really aims to serve as a foundation at some point to implement this type of analysis in the Servicio Navarro de Salud. The next example is going a bit beyond, so really trying to go for treatment suitability. And this is a collaboration we do with the Ipsen pharmaceutical company. And the project is called Emerging. So here, Ipsen and within the Emerging project, they aim to support the diagnostic and personalized treatment for patients with rare diseases. And they started with 2 different cohorts on rare cholestatic liver diseases and on severe short statures. And we plan to expand this. But basically, this type of disorders have treatments based on which part of the pathways are affected. So a treatment might not work for all patients presenting with these diseases. And this is the workflow that we are doing. So first, they recruit patients in Spanish hospitals in Spain. Here, it's basically hospitals that do not have access to whole exome sequencing themselves. They do extraction of the sample. Then they extract the DNA at Mar Genomics here in Barcelona. And at CNAG, what we do is exome sequencing, data processing, and we look for SNVs, indels, and CMVs, and we make the results available in another instances of the GPAP. Then we do variant prioritization and interpretation, and we send a scientific report with a turnaround time of 4 weeks. Then these results are sent directly to the referring clinician. So here, the pharmaceutical company do not have any, is not involved in the whole process. And also, we provide the raw data and access to these results to the hospital so that they can look at the data themselves if they are interested in. So far, 14% of the cases are eligible for treatment, and this is a good result for them, meaning that now they have a diagnostic, and together with this diagnostic at the same time, they also have access to that treatment. So we avoid this possible second odyssey on trying to identify which is the better treatment for those patients. And also, even for the negative one, it's a result, because this means that they would not have responded to this type of treatment, so they can look for other approaches without losing time in a treatment that would not have worked. And in this sense, at CNAG, we are also developing the Tritabulum database that Yanis also mentioned before, that was started within Solvardi, and we continue within Herdera. So this is work that is led by the INSERM. But basically what we do in this database is to try to link variants and genes to existing treatments, so that when a geneticist identifies a diagnosis, they can go to a database and try to find if there is already an existing treatment, so that we don't lose time with this second possible treatment odyssey. And this is the last pilot, the last example. So we've seen how we can improve diagnosis, how we can improve access to treatment. And this last example is really on how to find a diagnosis before even having symptoms, so we can prevent maybe the disease initiation or progression. And this pilot is done under the Screen for Care project, where they really aim to improve diagnostic. And there are 2 different approaches. Today I will focus on this genetic newborn screening. So basically here we have 3 different levels that we are going to test. The one that we call TREAT, which is to create a panel of treatable disorders to screen newborns. The second one is the ACT, so a panel on actionable disorders. And the third one is for symptomatic newborns to do whole genome sequencing. But I won't enter into this now. So I will basically focus on the work that we are doing for the TREAT panel. So this means that by newborn screening to do this panel of 245 genes. Here you have a list of the type of disorders that are involved in this panel. And basically what we aim to do is to screen 20,000 newborns across Germany, Italy, and France. And then they will recruit these newborns. The sequencing will be done centrally in Rome. And at CNAG and Genomics, what we'll do is the analysis and filtering and prioritization of pathogenic variants. Then we will issue a scientific report. We expect 98% of cases, around 19% of cases, to be negative. But for the ones that are positive, then they will go, we will send this information directly to the clinical site, and then we go under the diagnostic workflow, meaning that they will get new samples, they will also test the parents, and they will issue a diagnostic report. And there is also a way for variants of uncertain significance that we are sure we will encounter, and those ones will go through the European reference network under the umbrella of the Herdera project. So basically, conclusions for this pilot is that in the coming years, it will be crucial to assess this type of approaches and the added value of genomic newborn screening. This is not the only project that is currently ongoing, and indeed there are several at national level, like in Belgium or in France, that will be starting. But really to understand the addition of the plus of doing this type of approach. And here, apart from the technologies that we think are already in place and that we can do this type of approach, we really need to take into consideration the ethical part, because this is really, it will remain central, and we cannot, even if the technology is there, we cannot continue without taking this into account. And also taking into account that it's a country-based decision, because each country has different regulation towards, for example, applying whole genome sequencing for healthy newborns. And these are the general conclusions of these different pilots. So we've seen that genomic technologies and methodologies are improving, but are there, and they really help to accelerate and diagnosis and this access to treatment. That we need secure and robust infrastructures paired with also the strong expertise, not only in these technological centers, but also at the clinical site, to really have this multidisciplinary collaboration, so that we ensure that we can translate genomics into clinical practice. And finally, genomic centers such as CNAG can really play a role in having these pilots working, and also in this knowledge transfer to support the adoption of genomics by healthcare systems. And this means to really take into account the regulatory part, and as also Yanis was mentioning, this is clearly something that will be done in Herdera to facilitate that research goes into clinical practice. And this will really promote equitable access to disease for prevention, diagnosis, and treatment. And that's the last slide, just to acknowledge all the work from many, many people here at CNAG, and more, or at least more specifically to my colleagues from the bioinformatics unit that is led by Sergi Beltran. And thanks for your attention.
Do we have any questions? No questions. Did you understand? Ah, yeah, you did. No, it was very clear. Thank you very much. Good. Thanks a lot. Good. The next presentation is from Christophe Fleury, who is from Oxford Nanopore Technologies. Just do that. Just go there.
Perfect.
Thanks.
Right. Good morning, everyone. It's a great pleasure to be here at CNAG and being invited. So I'm Christophe Fleury, and I'm talking for Oxford Nanopore Technologies, but I'm here today with Alfonso Troiano and Luisa Vigevani. So I'm not pretending that I'm going to cover everything. Please come and have a chat with us during lunch. When you work for Oxford Nanopore Technologies, there's one thing you need to do is to select, because there's so much thing, and I could talk about the innovations and what we have, etc., and you would be overwhelmed. So today I had to select few topics. Sorry for not covering everything. And you've seen the title. It was Rethinking Genomics and how Nanopore can change things. I want you to, come out of here with an idea that we can innovate in the applications that we provide, not only from a technological point of view. And you know very well about Nanopore in research and even in translational research, but it's a journey that we are starting to go all the way to deploying the technology to hospitals. And in my idea, a genomics technology should, at the same time, provide comprehensive insights. You don't want to do 10 tests. If you can have all the information in one go, this is what you want to do, then it should be rapid and flexible, so go against decentralization, so that you can, if there's one patient samples arriving, you can sequence and get the information. Should be bedside, right? You shouldn't have to send your samples in a centralized manner. And finally, should be cost effective, because it's great to have a genome that costs 200 or 300 euros, but if when you deploy to a smaller hospital it's 10 or 20 times more expensive, of course it can't happen. So first, about the comprehensive insights. You know that the genome and the variations of the genome is very complex, right? You have the SNPs, you have the indels, you have also, and more importantly, the structural variants, inversions, and also the CNVs. You also have the epigenome. And with SBS, short reads, you can detect very well SNVs and indels, and maybe you can get some CNVs. But you see that you're not going to cover even half of the variations. So is it surprising that in the case of rare diseases, 60% of the cases remain unsolved when you look only at a little bit of the genome and its variations? So with Nanopore, we can cover everything with just the Nanopore reads. We can cover all the variations and the methylations and genome modifications. How do we do that? I call that the corporate slide. You have to show it. So I'm not going to go into the details. Actually, Ivo could probably tell you more than me about all this. But essentially, it's in the name. We sequence DNA or RNA going through a pore, and we measure the variation of the current that we applied inside the pore and call the basis from there. What's more important here is actually the features of the technology and the benefits from the features. So first, we're going to sequence directly the DNA and RNA. Have you ever thought about this? When you use current technologies, you're sequencing a copy of the DNA, meaning you PC amplify it, and if you don't PC amplify it up front, you do it on the flow cell. Here, we're looking at the DNA itself, which means that we're going to be able to look at epigenetic modifications without chemical conversion. We literally look at them. We're also going to have access to the dark genome. Why is it dark? Simply because we can't copy it easily. The enzymes don't work very well. Here we're going to be able to look at it, and there are some medically relevant genes in this. We will be able to look at any size of DNA. We call it long reads, but it's not only long reads. If we have short reads, like cfDNA, we can sequence it and all the way go up to 100 kilobases. And finally, you see this current here. So we measure the current, we call it into bases, and then we're going to align it. And we're going to do that in real time, meaning you don't need to wait till the end of your run to have some results. And rapid diagnostic is really something for which Nanopore has a unique proposition. So what do you get from Nanopore through this pore? A classic 30x whole genome will provide you all this. First, the epigenome, as I said. You see here the methylation of the different bases. So we're looking at a classic genome browser view. We're looking at one region of the genome. So we see first the methylation status. Then we have access at the structural variants, as I was saying. You see there that we have this long deletion. It's about 6 kb, and you see that the reads are spanning through the deletion. For the ones who are familiar with these views, you see colors, and you see that we have 2 rows every time. This is the phasing. so we can say from which allele. We look at both alleles separately, and we see that the methylation as well as the structure variation, the deletion, belongs to a certain allele. And it's important because if you have one normal allele, and that's the one that is expressed, then that's fine. It doesn't really matter that you have this modification. And finally, we can look at SNVs as well, because we can do most, we can do the least as well. We have end-to-end protocols. This is the result of one PromethION flow cells, and you know that the PromethION 24, for instance, can run 24 flow cells in parallel, meaning 24 whole genome at 30x depth. And we are also very good at looking at triplet repeats, which is very important in some syndromes. So when you look at this, what do you get? Well, there's a recent publication by authors that have taken the cohort of the 1,000 genomes. This cohort has been sequenced by short reads about 10 years ago. They decided to resequence it with nanopore reads and long reads. And what they find is that, looking at just 100 samples, they found 24,000 structural variants. They didn't appear in the course of 10 years. They were always there, but we missed them. And a similar observation was done by Bater and collaborators. Decode, Decode in Iceland, looking at 3,000 Icelanders 2 years ago, they published that they found between 8,000 and 10,000 variants that they didn't detect with short reads. Their effort is continuing right now to try and unravel those variants that we've never looked at. We didn't know they were there. And this is why it's urgent that all the national, transnational efforts that look at cohorts sequence, use nanopore reads in order to understand, to look at the whole picture of the genome. Because this is how we're going to be able then to feed that into the clinical practice. You see here, they mention also methylated regions. And you may know that a few days ago we announced a big partnership with UK Biobank to look at 50,000 samples, the epigenome in particular. There will be genetics information as well. I don't know what they're going to do with it. But all this is show you the trend that exists in genomics today. We're looking at the whole genome and all its features. Now, I was saying, we're finding all this. Does it matter? Well, that's actually you, the scientists, to tell us if it matters and how it matters. But there's already few studies like this one from United Arab Emirates, UAE, that have looked at some particular pathologies and showed that by using long reads, nanopore reads, they were able to detect 13% more or increase the diagnostic yield by 13%. So it matters. They could resolve cases that they were not able before. Now you might think, well, 13% is not yet, all the way to 100%. But this is because the translational work hasn't been done yet. We need to look at more cohort and look at more patients in order to, know exactly what's going on or what happens. Right, so all this was for whole genome, and I understand very well that sometimes you can't do whole genomes. You still need panels. So we haven't been very much in the panel world before, but we're starting. And panels is usually using an enrichment method, right, to look at only few genes. And we have a partnership with Twist Biosciences with a pharmacogenomics long reads panel. But have you ever dreamt of being able to do enrichment without going through the painful wet lab capture process? What if you could just give a bed file to the software and it would do the enrichment for you? That's the dream, right? If you've done that in the lab. Well, this is what adaptive sampling is about. I'm not going to go into the details of the technology because I only have 15 minutes, but we can prepare a whole genome library and then tell the software, thanks to the real-time analysis, what sequences they should look at and sequence and which ones they should disregard. And we have an inherited cancer, comprehensive hereditary cancer panel coming later this year, 241 genes. And we have also a pharmacogenomics panels with a bit more than 150 genes. And because we're looking at native DNA, we're going to be able to look at the methylation, if that matters, structural variants, but also, thanks to the long reads, be able to map the pseudogenes and the CNVs more accurately. So mentioning cancer, And mentioning partnerships, we also announced a few weeks ago a huge partnership with Genome England regarding several programs. But we've been working with Genome England for at least 3 years. And they have a rare disease program and also a cancer program that they call Gel Cancer 2.0. And their objective is to assess the utility of the technology for clinical applications. And obviously, we had a talk this morning about leukemia. And for all these disorders, the gene fusions, the gene translocations are very important, and long reads benefit fantastically from this, can benefit fantastically to the detection of those gene translocations. And this is what GEL is doing. And they claim, you see at the bottom, ONT technology is clinically promising. But there's also another feature. So that's for the comprehensive insights, right? But what about the rapidity and the bedside side of the technology? So I don't know if you're very familiar with central nervous system tumors or brain tumors. To classify them, you need to classify them because depending on what type of tumor it is, the surgical act as well as the treatment will be different. So currently what they do, they do the classification based on methylation, and they use arrays to be able to detect or to classify the tumor. You see there, it takes about a month to get the result. So the surgeon needs to, and you know, the brain tumor is not easy to access, so they need to operate and get the genomics results, and then, you know, decide what they're gonna do. And there's a group. There are several groups actually. Some in Germany, Netherlands, and this one is a publication from a British group. They have thought, well, and what about we leverage the fact that we can analyze nanopore data in real time to do the classification as the surgeon operates. And that's what they did. So the surgeon takes a piece of the tissue of the tumor, send it to the lab that is next door, right? They put literally the sequencer in the room next to the operation theater. And then they prepare the DNA and the sequence. And you see that in 2 hours, 1 hour and a half of which are extracting the DNA and preparing the library, but in 30-minute sequencing, they get a profile, methylation profile, that is enough to determine what tumor the patient has. After 2 hours, the operation is not over, so they can go to the surgeon and tell them this is the type of tumor there is, and they can make a decision, an educated decision. And they usually let the sequencing run carry on, and they get even more information, CNVs and other mutations, because why stopping the run at this point? So this type of rapid approach benefit not only from brain tumor, but also pulmonary infections. Sometimes that can be very serious. NICU PICUs, or neonatal intensive care units. And well, again, leukemia. All these, they achieve results in less than 10 hours. So I see my time has nearly elapsed. Sorry. Anyway, so there are several instruments, right? But what I want you to focus on, and I'm going to take the laser for the first time, is I was talking about cost effectiveness. You see here, these are the PromethION flow cells. So here at CNAG, you have access to the P24. There's a quite big sequencer. The price per gigabase is around between 5 and 7 euro. But of course, it's available for the big instrument. But you see this instrument here? This is the P2i. So it's that big, deployable, right? You get the same price. So the technology is affordable, and you get the whole genome or a panel and the methylation as well for this price. Anyway, I had to have one slide about innovation. The sequencer is one thing, but actually everything, chemistry, sensing, happens on the flow cell. The real technology is there. And we are able to increase the output of the flow cell independently of the change of the instrument, meaning that when you invest in an instrument, you're going to get, hopefully in the future, more and more data. So the price will go down, and the capacity of your instrument will increase. And you see there, this is currently what you have. So you get about 100 gigabases per flow cell, but it's with the few improvements that we're doing, and some are being tested in the field, you're getting more and more data. So same instruments, but more and more data, and the price going down as well. So rapid conclusion, we have several workflows. I didn't talk about single-cell RNA-seq. We can do that. I didn't talk too much about 16S. We can do that, etc., etc. We're partnering with a lot of people for the tertiary analysis because we're good all the way to the secondary, but you know, you need professionals after that for analysis. And we're partnered with Fabric, GeneYX, Varsoms, and a few others. And also, we are working towards making our sequencer more clinical, meaning trying to provide you with a stable environment for at least 2 to 3 years, so you don't have to revalidate the test over and over again. Stable environment means stable software that we don't update, stable regions that we don't change without notifications, etc., etc. So thank you very much. London Calling 2025 is coming. Now that you know why you should use Nanopore and that you should use Nanopore, please generate data. Ivo, I'm sure, is happy to do the sequencing. And you submit your abstracts. Thanks.
Thanks so much, Christophe.
Can grab me at lunch.
Yeah, you can grab Christophe at lunch. And now there is the final presentation before lunch, which is Agatha Schlüter. She says she doesn't know how to say her own name. I just probably said it correctly, right? No, she didn't say that. She says there are people who know how to say my name better than I can say it. So what I should say is in the initial program there was Aurora Pujol, but unfortunately she had an accident last week, and so Agatha is going to be a very good stand-in for her.
Thank you. So thank you very much, Ivo, for inviting us. So inviting Aurora. As you said, Aurora had an accident last week, but she's fine, but it was not possible for her to come in today. And so I'll do my best. First of all, I would like to thanks to CNAG, because all this work has been possible thanks to CNAG. And so we are very grateful to your work. And I'm going to explain about our tool, that is ClinPrior, that it allows us to prioritize genetic variants for undiagnosed diseases, leukodystrophies, spastic paraplegias, and ataxias, and in severe COVID conditions. Also, thanks to this tool, we could discover 12 novel diseases. And at the end, I'm going to explain about disease 1 as an example of what's next after genetic diagnosis within a treatment opportunity for this disease. So ClinPrior has been published in Genome Medicine, and as you can see, there are some CNAG members in this publication, like Gemma Bullic and Sergi Beltran. And how does it work, ClinPrior? So ClinPrior needs 2 inputs. One is the phenotypic description of the patient in HPO terms, as Leslie explained before, is a standardized clinical. Is a way to standardize clinical information. And the second input is the variant call format file from exome and genome. And first, the ClinPrior, what it's doing is calculating a phenotypic association metric, taking into account the HPOs from patient and comparing against the HPO, the gene HPO associations present in a multi-layer network. And also this metric propagates through a network built with physical protein-protein interactions and functional protein-protein interactions. So thanks to this propagation, we can reach out new candidate genes that are adjacent to disease genes that are described or similar to the phenotypic as our patients show. Taking together the HPO, or the phenotypic description of the patient, and the variant call format file, ClinPrior can prioritize the best genetic cause that could explain the clinical of our patient. So we wanted to evaluate the performance of ClinPrior, and that's why we perform a benchmarking using first a synthetic cohort. This synthetic cohort was a chimeric gold standard exome plus a pathogenic ClinVar variant. And then we evaluated this performance. Here you can see the display in AUROC presentations. The AUROC is calculating the area under the ROC curve. And you can see that for known genes, we simulate the patient clinica using the gene HPOs associations present in the ClinPrior network. And when simulate the patient clinica with these HPO gene associations, we get the best result, no? That is close, an area close to 1. But when we simulate the clinica with random HPOs, then we are getting down the AUROC result, meaning or underlying that the retrieving HPOs and the inaccurate HPOs describing the clinical phenotype is a very important step to get a good prioritization. But also we wanted to evaluate if ClinPrior was able to prioritize candidate genes, meaning genes that are not previously associated to disease or gene disease. And that's why to simulate a candidate gene, we remove it from the ClinPrior database, the gene HPO associations. And in this way, simulating again the patient clinica with the gene HPO associations, we observe that the AUROC is getting lower if we compare with a known gene. And in this case, ClinPrior can reach to the candidate gene through the propagation, through the network propagation. Also, when we simulate the patient clinica with random HPOs, then the AUROC is getting lower. And in this situation, ClinPrior can reach out to the candidate gene only through the variant predictor score. But so it was for a computational cohort, no, a synthetic cohort. How it works in a real-world cohort. So first, we used a cohort with 135 patients with a spastic paraplegia and or cerebral ataxia, and we apply a pipeline that is most commonly used for everyone. That is first trying to find out the pathogenic and likely pathogenic variants, single nucleotide variants from exome sequencing. But after reanalysis and most important, functional studies of variants of uncertain significance, we can increase the diagnostic yield. And then when even this diagnosis was negative, then we could improve the diagnostic by sequencing by whole genome. And so ClinPrior helped us to diagnose this cohort, but only could be involved in the sequencing or in the analysis of exome and genome sequencing single nucleotide variants. And we used another cohort. In this case, is a cohort with 126 patients, and in this case with leukodystrophy disease. Again, applying the same or similar pipeline with exome reanalysis, functional studies, and genome sequencing, we observed that we obtain a similar diagnostic yield, close to 70%. This is a very high diagnostic yield. You can see that is doubling what is provided by service, diagnostic service. The main reason is because we can provide functional validation. That is very important. And as well, what it was important as well is that we were able to discover 12 novel identities, 12 novel diseases. Diseases, here in this display, you can see some of the 12 novel diseases that we could describe. After, at the end, I'll go with in more detail with DGIS1, this enzyme. But we're not only novel diseases. I think that it's important to take in mind that there are also phenotypic expansions, meaning that are genes associated to disease, but we can find phenotypes that doesn't match perfectly or doesn't fit perfectly with what it was described. And so it's what we call phenotypic expansion. This is the case for the GFAP. This is a gene associated to Alexander disease. It's a children disease. What we found in a family with 6 members is that they found a different clinical and cellular phenotype with a larger cells that were not previously described in Alexander disease. But there are more phenotypic expansions described in our cohorts. And in conclusions, maybe we can say that 7% of our cases showed an expansion phenotype. So take into mind that this is very important to consider for the diagnosis. And also novel inheritance modes. No? You cannot discard because it's not matching the inheritance mode. So, for instance, we found for a potassium channel subfamily gene, we found a patient that instead of a dominant inheritance mode showed a recessive, and as well for a serial tRNA synthetases, it was previously described only recessive, and we found a new dominant inheritance mode with different also clinical. And also now we move to another field. We wanted to participate or to collaborate with a grain of sand in the COVID time. No? That's why we joined to the COVID human genetic effort, led by Jean-Laurent Casanova in the Rockefeller University. And thanks to easy genomics, we could sequence patients infected with SARS-CoV and find susceptibility genetic causes that driving the severe consequence of this infection. Interestingly, in this consortium, we found that through this consortium, that 15 of the severe COVID cases can be explained by an abnormality interferon production, and in 3% of this 15% is due to mutations in the interferon signaling, but the rest as well for the production of autoantibody against interferon type 1. So this is highlighting that not only genomics or personalized medicine is useful for diagnosing ultra-rare diseases or in cancer, also to try to stratify patients and maybe to be more prepared for next pandemic, no? And find susceptibilities in the population. And also through this consortium, we were also able to find the genetic cause for multisystem inflammatory syndrome in children that show mutations in the RNA sensing genes OIS 1, 2, 3. And at this point of the talk, reminding you that this tool, ClinPrior, is freely available, that you can find it in the GitHub as a package. You can provide BCF in 37 and 38 assemblies, genome assemblies. Also, you can download it as a Docker image and also convert this Docker image to a singularity, allowing to run this ClinPrior in HPC clusters. Limitations, so the most important limitations are the databases for protein-protein interactions and also for the HPO gene associations. The number and quality of HPO terms are very important to prioritize properly the genetic cause of the mutations. For the moment, only prioritizes single nucleotide variants, but in future upgrades, we plan to add structural variants in the pipeline. Integrate additional variant damage predictors like a splice-I. Also include other omics like RNA-seq in the prioritization function. And as an example of what's next after the genetic diagnosis, so we could diagnose disease 1 as a new candidate that causing hypomyelinating leukodystrophy. So you can see in this interactome that Klinefelter could reach disease 1 because it's a close interactor of other enzymes causing leukodystrophy like GALC and ACER3 that are also associated to the leukodystrophy diseases. Disease 1 is an enzyme that produces ceramide from dehydroceramide. This enzyme is present in the sphingolipid pathway, and as you can see in this pathway there are more enzymes associated to disease like CRAVE, I don't know, like CRAVE, Gaucher, Niemann-Pick. And why? No, because the sphingolipid pathway is very important, not only for the production as a fuel of sphingolipids or structural lipids, as well because it's very important for the signaling of the cell. So patients with mutations, carrying mutations in this enzyme show lower levels of the product of the ceramide, and also show higher level of dehydroceramides, the substrate. So with an impair, obviously, of the ratio substrate product. It was reported that the fingolimod, that is a drug used for the treatment of multiple sclerosis, can inhibit the previous step, can inhibit the enzyme of the ceramide synthase. So blocking this enzyme, maybe we can reduce the accumulation of the dehydroceramide, and that's why we are planning, or we are wondering if maybe a substrate reduction therapy could ameliorate or improve the clinic of our patients. For this purpose, we made a zebrafish model, a disease 1 knockdown, and after fingolimod treatment, we could find that we could rescue the phenotype of the model. We can observe that we can partially normalize the levels of the dehydroceramides and ceramide, and we could also rescue the locomotor and the myelination phenotype. Also, by untargeted lipidomics, we could find several biomarkers very powerful, and that were able to distinguish the patients with this that are with problems in the synthesis of these fatty acids, no, these sphingolipids, with a very high AUROC, higher as 90%. Taking all together the promising results in the zebrafish model and the identification of these biomarkers, we apply for an orphan drug designation pilot trial with the use of fingolimod treatment. And then we could recruit 4 patients, 2 from India, 1 from Barcelona, and the other from France. And after treatment, what we observe is that for the patient 24, that is the blue one, they could partially normalize the levels of the dehydroceramides and the ceramides. And most importantly, also the NFL and the GFAP, that are neurological disease biomarkers, that are higher in this disease. After fingolimod treatment, we could, in patient 24, rescue partially or reduce these biomarkers levels. So why is working so well in this 24 patient? 24 patient is the youngest one with 1 year and a half, and after 9 months of treatment, we observed that in the MRI, signal was improving, and also in motor or locomotor and cognitive scales also was improving. After these promising results, we are planning to include 10 more patients through the GLIA consortium in USA, thanks to Adeline Vanderberg. And so at the end of the talk, what we have learned that, first of all, we have to trust our clinician. The clinic is very important. It's important integrate clinical genomics, but as well perform functional validation for BUS, for variants of uncertain significance. That applying integrated genomics, we can reduce the diagnostic odyssey from 10 years to 8 months, open our mind and take into consideration that doing exome and genomes, there will come new atypical cases with phenotypic expansions, complex phenotypes, dual diagnosis, also working team and network. Thanks to GeneMatcher, we were able to find several patients bearing variants in the same genes that our patients. And the diagnosis is not the end, it's the beginning to find a cure to our patients. And so, as a perspective, we are happy to be involved in the SIDS ALS project. And together with Anasteva, we are planning to find new gene in the family ALS cases that are still negative, and also in collaboration with IMPACT Genomica 2. So thank you very much for your attention, and thanks to our group members, to the patients that gave us the samples, and also to the neurologists and neuropediatricians that supervise our clinical job. Thank you very much.
Thank you very much. Do we have questions? You're not very talkative today, guys. but I have a question for you. You know, you said that you go from exome sequencing to reanalysis to functional examination to whole genome sequencing, and you add a little bit in all of those steps. If you reverse the order of things, for example, the functional and the whole genome sequencing, are there things that you can explain out of the whole genome sequencing that makes you not having to do the functional validation?
If I could choose, first I will do the whole genome sequencing. And with the whole genome sequencing, then you will have deep intronic variants, but also structural variants, and then perform the functional validation, of course. It's only a matter of money.
Okay, good. So we have to make it even cheaper. Yeah. Okay, good. Okay, thanks ever so much. Then I believe there is food outside. Go to all of the tables at the back. Don't stand at the first table because there will be quite a lot of people, and just go to where you still see food and start there, and we fill up. Roughly 1 hour of lunch.
When I explain to you what we've been doing and why we've been doing what we've been doing in child psychiatry, and more particularly regarding autism spectrum disorders and the search of clinical phenotypes that really help us to understand the biological causes. So these are my affiliations and funding. I mean, in fact, these are the affiliations and funding of the whole program, and the conflicts of interests related to this talk. So I'm assuming I'm in a heterogeneous place now, people that really know a lot about genetics or are interested on genetics, but probably not everyone is focused on autism spectrum disorders, although I see some people that I know is, but probably not everyone. So the very first thing I want to do is try to let you know through what I'm going to talk about, what's autism spectrum disorder and why it's a very heterogeneous condition. So when we talk about ASD, we are talking about clinical condition in which we need to have these two domains with severe or at least with non-affectation. The domain of the social communication, so it's not only about having deficits, but also about having difficulties or having deviations from what is considered normal or mean, and restricted interests, repetitive behaviors, insistence on sameness, and some sensory sensitivities, hypersensitivities. When we have this, and when this appears early in life, it used to be said that it had to appear before age 3. Now we don't have that, that borders, but that should happen in early infancy, except some cases that are not as severe, that although most symptoms appear before, do not really show up until adolescence. When we have that, and when there's also problems in functioning that are derived to that, we are talking about ASD. The problem is that ASD alone is very, very uncommon. What we know is that many times we also have problems within the intellectual disability spectrum. It's not every patient, but many of the patients we see at least do have some mild or borderline difficulties, or it's not compensated within the different domains that we measure when we measure IQ. There's problems with language skills, there's problems within adaptive functioning, and it's, again, heterogeneous. Some people will need lots of support for their daily life, and some other people will not need support, probably for most of their activities. And also, we have very frequently another expression of illness, which are the psychiatric symptoms or even illnesses such as ADHD, anxiety, depression, OCD, or some symptoms that sometimes are very prevalent, are very important, and very disabling. And on top of that, many times other problems that are not linked to psychiatry, but that have a lot of impact, such as high prevalence of epilepsy, gastrointestinal diseases, and motor problems. So this is the picture. It's very difficult just to have one person with autism that is exactly the same than other person with autism, right? But what we know is that people with autism are people that have neurodevelopmental disorders. And when we talk about neurodevelopmental disorders, we are talking about conditions that appear early in life that really have impact in different expressions of brain development. And we can study them starting from the bottom. We are able now to find some gene problems that we are going to talk about today that also affect the proteins and the cell and the circuit and formation, and at the end, the phenotype. What we can also do is start working with the phenotypes and go top down and trying to look for what are the genetic causes. And this is what we have been trying to do within our team. We know that it's not everything explained about genetics. There's lots of room for the environment, but when we talk about the environment that we are interested in, is really the environment that has to do with the intrauterus environment. So this is the environment that we really need to look for. Nevertheless, what we consider the outside environment or the educational or relationship environment has lots to do with the comorbidities within autism. So really, at the end, the expression of the phenotype sometimes also depend on other conditions. And this is very relevant because many times medications that we use so far, these are not for causative mechanisms, but medications that we use so far really have effect on the comorbidities. And we need to have that in mind. I'm talking about a condition that is really new. So the first definition of autism, the first definitions, because it's a disease that was really known for the first time or named for the first time in 1943 by Kanner, and next year in 1944 by Hans Asperger. Really, we've got a long way since then. So for the first years, the first 20 years were devoted mostly for description, for really enlarging the numbers of people that were people with autism. From there, what was done was mostly trying to organize a diagnostic system and try to start working within potential treatments. Very important aspects having to do with the cognitive affectation were described afterwards, and it's been within the last years, the last 7-8 years, that we've started to work on genetic and model research. So it's definitely quite novel and quite new, and this is what I'm really going to focus today. We know that it's not only with autism, but with other psychiatric disorders, there's an increase in the relative risk within family members. And this increase in relative risk is not only due to genetics, but also due to the shared environment, but mostly to genetics, of course. Here we can see that autism, the relative risk is 8.7, so it's the highest, followed by bipolar and by schizophrenia spectrum disorders. But we know that most genetic risk for autism resides in common variation. So although what we know more as per now has to do with de novo genetics, genetic causes, as happens with other psychiatric conditions, most genetics comes with common variation, particularly in those cases with no intellectual disabilities. For those cases with intellectual disabilities, probably the proportion is geared towards more problems in de novo variation. So when we talk about that, when we talk about rare variation, we've learned a lot within the last years, and now we know that there's about like 150 genes that can be here as potential causes of autism. Here you can see some of them with the highest penetrance, but it's possible just to make panels for autism spectrum disorders. Of course, the findings are not always exactly the same in different studies, so different studies can yield slightly different results regarding the genes that are like really proposed as causative of autism. But interestingly, there's an overlap between genes that are putative causes of autism and those that are related to intellectual disability. So the developmental disability genes are marked here in red, and if you see those that really overlap between different studies, most of these ones are also related to developmental disabilities. So where are we now? We've got lots of potential genes that may be affected or maybe the cause of these very few phenotypic expressions when it comes at least to the core symptoms of autism, not that few when it comes to the whole expression, including all the comorbidities I mentioned before. But in the middle, we also have another convergence mechanisms. So it's not only about genes, it's also about what those genes really make, what are the functions of those genes. And we know that these genes, some of them are related to transcription regulation, neurodegeneration, synaptic transmission, or scaffolding proteins or genes related to chromatin. And there are not many mechanisms so far that have been identified, much more genes than potential mechanisms so far. So with that, we've been trying to work on patient stratification, this wide array of people with autism that we see in the clinic, trying to find at least common mechanisms or ways of subgrouping these people to look for more homogeneous phenotypes, genotypes that allow us to implement particular interventions that may help with rescuing some symptomatology. This is one of the projects I want to present to you. This project is still ongoing. It's an IMI-funded project through the European Commission, the AIMS2 trials. It had 2 steps. And in this trial that had different PIs in Europe, also has another brother in Canada now, where they are developing similar intervention studies that we've designed. What we've been doing is trying to reach to agreements about how to measure those aspects that we know that are central, are core in autism, such as cognition. So one of the things we've been trying to do is not only measuring symptoms in the same way, that's also is more or less structured, but trying to reach to a definition of difficulties within cognition that we see in this patient with different tests, including not only pen and paper tests, but tests such as tests with eye tracking. We also try to evaluate brain function, trying to look for differences in differentiating subgroups with MRI and EEG, as well as using genomics and biochemistry to make subgroupings. So the idea is try and reach to a very big cohort in 3 countries now, just to make sure that we are measuring things the same way. Now we've put together another project, and this is a project that's also still ongoing, and this is funding from the Spanish Ministry of Health and Science and Innovation. I think that is the personalized medicine call. The last call was this year. I think there's not going to be more. And this is like it's a project that was funded 3 years ago. In this one, that's also a a project in multiple sites. We have clinical sites. We have sites that really work mostly with statistics and with genetics. What we've been doing is organizing the work in 2 ways. So on the one hand, we've been trying to put together all the cohorts that these groups already have. There's many people doing work on autism, but so far people were doing this work separately. So we are putting together the cohorts and trying to include more people just to reach a significant number of 1,000 patients. We've included also, we're trying to do also integration of the previous samples that we have. Many of these projects have already been collecting not only genetics, but other biomarkers, and we've been putting together that. So from that, what we are going to do is doing genomic sequencing, and this is the work package that probably you're more interested on that, and proteomics, trying to prioritize what are the variables that we need to do Sanger confirmation afterwards. We will try and develop some diagnostic algorithms based on our findings, and we'll also aim for doing algorithms based on cost efficiency, because the interest of the project is to have like clinical transference afterwards. So this is the idea in sum, just to have a big cohort. Now we've recruited like almost 700 at this stage to do a deep phenotypic characterization, all this wide array of clinical manifestations I mentioned before, to have exome sequencing in every participant, but genome sequencing at least in half of them, and we are now prioritizing which ones are going to go through that, doing the cost efficacy algorithms, and also making algorithms in diagnostic decision. We've piloted some data so far, and this is the data from the first patients that we included in our site. And we've been doing modeling in clusters. To do so, what we've done is putting together, for this very first example, putting together clinical characteristics and inflammation data. And what we've seen is that within 4 clusters, this is what the model came out with, we saw differences in age, in some of the inflammation parameters, in the presence or absence of regression. And regression means whether the person were developing the milestone, even if not at a normal pace, but at least with no loss of functioning. And regression means losing the, not regression is doing that normally, and regression means loss of functioning if in any developmental domain. What are the age at which symptoms were more evident? What's the repetitive domain measured by the ADOS, which is the gold standard in diagnosis, and what's the IQ? So including all of that together with the inflammatory the inflammation data, we came out with a very interesting cluster that's composed by patients with regression that's enriched in women. I didn't mention that before, but as happens with other neurodevelopmental conditions, autism is far more prevalent in men than in women, and in boys than in girls. So it's not really depending on age. We see that the proportion, if we take the whole sample, national databases, is around 4 to 1, male, female. If we go to particular subgroups, such as older women with no developmental disabilities, it's 9 to 1, right? So for this cluster, we see that's enriched in women with age of onset 1 to 3 years, with severity, important severity of autistic symptoms, but not that important severity in developmental delay. So the borderline IQ, and importantly, these markers of innate immune activation. And also that's another interesting thing, that when we looked at these patients, we saw that some symptoms that are these collateral symptoms that come together, such as dysregulation, are very prevalent in these people. I'm not saying this is included in the model, so it doesn't have the same value, but interestingly, these patients were functioning this way. So why we want to go this road? What do we expect from working in genetics in such population? This is an interesting example of personalized treatment within autism spectrum disorder. And this is one interesting example of one of the genetic discoveries that happened relatively recently, which is the Phelan-McDermid syndrome, the 22q13 deletion syndrome. So this deletion affects the shank gene. The shank is a gene coding for a postsynaptic structural protein that what makes is that scaffolding between the glutamate receptors and the intracellular signaling that allows the excitation through via glutamate to happen. So it's very important in synaptic maturation and stabilization, and when there are problems, there are deficits in the excitatory transmission. So for these models, there's been some developments in potential ways to rescue the phenotype. I'm going to focus in the IGF-1. There's been some work done in neurons that are derived from pluripotent stem cells in humans. In human patients with the Phelan-McDermid syndromes, and treated with the IGF-1, the insulin factor, that restores the excitatory function in the neurons, right? We can see that also within knockout mice for the shank. We see how after treating, and this is very, very, short term, it's 2 weeks of treatment with insulin factor 1, the restoration of the AMPA activity, And there's start to have to be some data out there with patients. This is a very still underpowered study with patients with the SHANK3. These are mostly early ages. And here you can see that there's some rescue in the social withdrawal. That is one of the social deficits that we can measure with the ABC scale. And particularly important, the rescue in the hyperactivity. The rest of the findings are not really significant. Taking it from there, what we've been doing in our institute is trying to use this for particular cases and studying these cases. We are not doing still cohorts, but studying those cases as deeply as possible and also publishing the results. So this is the case of a person who is now 22 years of age that has the Phelan-McDermid syndrome. She came to the clinic at age 9, and by age 9, one of the mother of this person is the president of the Phelan-McDermid organization, and she really pushed us to try this treatment. So we are now working in other patients doing the same. We started treatment, and here you can see how the social responsivity scale goes up and down, whether patient is on, off the medication. We've been measuring as deeply as possible not only this aspect, but also different aspects related to behavior that many times come together with autism, such as agitation, withdrawal, stereotypic behavior, hyperactivity, or repetitive speech. And although hyperactivity, as we saw before in the data I showed from the clinical study, is the aspect that really fluctuates the most, we can also see some changes, mostly in repetitive speech and agitation. This is the social responsiveness scale, and we can also see that in the total, there's worsening when patient is not with the medication. Going up means worse aspects related to social cognition, and improving when the person was on the medication. Right, so this is just a resume. And to finish with that, and this is not a finding, or is related to a different neurodevelopmental condition, which is schizophrenia. Most cases of schizophrenia start before age 35, and most of them, although there's also a neurodegenerative aspect related to particularly those starting a little bit later, but most of them have deficits in neurodevelopment. So using polygenic risk scores, we now start to be able to find different clusters of people, and in this case, I'm talking about responsive response to medication. So we're this is like very preliminary data, but being able to differentiate those that are responsive or not to medication, and within those that are not responsive, we can even see differences between those that are ultra-resistant or those that are resistant even to the medications that are used for resistant schizophrenia that is closer. There's some preliminary data also on being able to differentiate those that are patients with more or with less suicidal risk. So just to finish, a couple of slides to put in place why it makes sense to work with genetics within this population, even knowing that we are not as advanced as in other areas of medicine, as probably you are in your different areas, right? So why does it make sense? It makes sense because we start to understand something about the associated comorbidities, and this is not unlikely that we have findings related to other areas in the patients that we are now analyzing. So this is absolutely relevant for these people, also considering that people with autism very likely die or really have high morbidity for illnesses different than autism. They don't speak, they don't communicate pain, they really have a struggle going to the hospital. So this is an important aspect. Definitely, if we can identify clusters of people that are going to regress, that are going to have worse prognosis, that's also very relevant, because probably the strength of the work we put, now people just do behavioral therapy with these kids, right? So really advising on whether this is important or not, whether you are going to be losing your time and you should be doing another thing is also absolutely relevant. We really think that pharmacogenetics is a very interesting avenue, and I'm sure that there's going to be something now being said in the other presentations. And definitely one of the things that matters most to parents in this case of patients has to do with inheritance. So people want to know whether having another child with autism is also going to carry high risk. I mean, whether having other kids are going to be problematic, and will they have high or low risk of autism? And definitely knowing the cause make people change their mind, and many people still think that the cause of autism is that they are bad parents, bad mothers, that they didn't do the work well. Definitely, by now, we can know that around 20-25 of genetic variants are causal for autism. This is much more than doing MRIs and doing metabolic tests that so far has been the standard for the diagnosis of the cause of autism, right? So we expect that using genetics allow us to improve medical care, to understand better the prognosis, for many people to find peer support. There's many associations that start to grow, and people really find very helpful understanding what other people have been through, to modulate the interventions that we are carrying out, to deal with the expectations as soon as possible, and really not to lose time and effort on treatments that are not going to work, to facilitate research on mechanisms and potential interventions, to make reproductive decisions, and definitely to explain to our patients and our parents what's the potential cause of the symptoms they see in their everyday lives. This is part of the team we work with. This is the team of the Child and Adolescent Psychiatric Unit. And that's it. I want to thank you for your attention today.
Do we have any questions? Yeah, Pedro.
Yeah, so I was interested about this cluster you mentioned of patients with sort of regression and signs of... Sorry. I was interested about this particular cluster of patients you mentioned with regression, signs of inflammation. I mean, is that actually autism, or could be that it's another sort of something disease that mimics autism?
You're saying that why?
No, because I mean, it's not something that I typically associate with sort of inflammation in autism. So that's why I was interested in this particular cluster.
Yeah. So it's not that the rest of the sample did not have high levels of inflammation, because this is a constant. We've seen that, right? But in this cluster, levels are higher. And really what we see there are patients that are much more affected in terms of autistic symptoms. So they are not that developmentally delayed, but they have more severity of each autistic symptom. And they also have regression. And regression meaning starting to talk and regress in talking, mostly talking, much more than other things, and social. So we didn't include here comorbidities. We just look at that on the top, what I mentioned about like instability, but we also know that this is very common, and particularly in those patients that are girls and that are patients that do not have developmental delays, gross developmental delays.
Okay, thank you.
Okay, thank you very much, Carmen. So the next presentation is given by Yolanda Sanchez, who comes to us from Agilent Technologies.
Okay, good afternoon, everyone, and thank you very much for your invitation to this great symposium. Today I'm going to show the genomic solution that we have in Agilent Technologies. Just to remind, the workflow of to the NGS preparation library, we have some different steps that included sample preparation, library preparation, sequencing, and data analysis. Today I'm going to very focus on sample preparation and library preparation. This is the most important step in the workflow to determine the sample that you want to identify in order to save time and money. You should do the quality control of the sample and also of the quality control of the library preparation in order to be successful in your sequencing. Usually, the quality control you can do manually via agarose or acrylamide gel for DNA or RNA, or you can do automatize in some instrument that we have in our portfolio. This allows to you to make the faster and very precise results. In Agilent Technologies portfolio, we have those instruments that allows to you to measure the quality control of the DNA and RNA. In order to get the size, concentration, integrity, and the purity of your DNA or RNA. This is very important in order to be very successful in the sequencing. In the Agilent portfolio, we have a lot of instruments that, depending on the application that you need, you can fit the instrument. It will work much better for you. This is the tape station system that is very well known for the NGS library and also for the genomics and also for cell-free DNA. This is a very automatized system that is very fast and very based on the screen test technologies. We have two different models. It's exactly the same, just depending on the volume and throughput that you can put in the same RAM. We have the fragment analyzer system, that is an electrophoresis capillary system that with the same application that tape station, and also it's included for the small RNAs. This is a system for much more high throughput and with much more resolution and sensitivity. And we have our femtopulse system with a very important application for the long-read sequencing and another application. This is a system very useful for a very ultra-high sensitivity level of detection. And we have also our bioanalyzer that many of know. This is a discontinuity system, although we are still keeping the reactive until 2030. So now, as I mentioned, our system is very fixed for the application that you want to read. And we have shown in many cases, in many studies, that our femtopulse system is very useful for the long-read sequencing on the PacBio and also in the Oxford Nanopore technology platform sequencing. We have these two application notes that can be available for you if you want to read it. as many of you know, in Agilent we are expertise on in the library preparation with our SUSELECT technology from more than 15 years in the market. We started with our SUSELECT XT technology that has been improved until now that we are in the SUSELECT X2, SUSELECT XTS2 that can measure DNA and RNA in parallel in the same process. Recently, we will launch the new SUSELECT Max for DNA and RNA that is coming soon. So basically, the improvement of this SUSELECT XTS2 is the turnaround time that is much faster, and also the hybridization and the cost effective is much more nice. So Agilent Technologies is very well known for the custom panel, that we are very specialized in the custom panel, but we have also a lot of catalog panels, like our well-known exome, that now we are in the version exome B8. We have a lot of another catalog panel, like clinical exome sequencing, exome B8 plus UTR. We have some mitochondrial genome. And now we are recently launched as a community design, we have launched the CRE4, that this is an exome that included some core exome B8 plus some low non-coding RNA sequencing and some clinical relevant regions. And we have also some catalog panel that include DNA and RNA targets, like this is our CpG cancer panel that includes some 679 genes for DNA and 80 genes for RNA that can detect a lot of mutations in a high variety of the cancers. So this is very nice, that could be applied in some clinical relevant projects. So our technology for SUSELECT can be prepared manually or can be done automatically in our systems. In Agilent, we have two different instruments that can be helped for the automatization. We have our system that is Magnus, where this is fully automatized, the samples. You just put the samples and just run, and in 8 hours, you will get all the library preparation ready for sequencing. This is a closed system for our technology, but we have our well-known Bravo automated liquid handling platform that is open for all of the library preparation technology that we can prepare in a very high throughput manner the library preparation. We are as I mentioned, our technology is open to be sequencing in all other platforms that are in the market. And now also, we are also doing some collaboration with the new platform for long-read sequencing, as PacBio and Nanopore technology that are in the market. Regarding the new acquisition for Agilent, in order for the target enrichment solution for liquid biopsy, I want to present Avida, that this is a solution that has in Agilent, that is a technology that has a very impact because you can get a library preparation from a very little amount of sample and no need to pre-cut to PCR, and you can do very fast hybridization, and you can put multiplexing in some sequencing platform as 192 indexes in the same RAM. So this technology is quite new because it consists of two different proofs that has a bridge and an anchor proof that can bind to the nature DNA and can make the enrichment on the target that you want to sequence in. So as a summary for the video portfolio, it's very important to remark that from very low input sample, as a 1 nanogram of cell-free DNA, you can do in parallel the DNA on the methyl target DNA, and with a very highly performance, improving the sensibility that allows to get a very high performance sequencing. And this is the catalog, the current catalog that we have. The current catalog that we have for AVIDA as a catalog panel that we have AVIDA DNA discovery and AVIDA DNA expanded, depending on the size of the cancer panel that you want to study. We have also the AVIDA DNA focus that are for some actionable genes. And we have the AVIDA methyl custom panel that is a custom panel that is a catalog panel that included a lot of targets for methyl DNA studies. So, and this is all that we have in the genomics portfolio for now. So thank you very much for your attention.
Thank you very much. Okay. Wait a second. Does anyone have any questions? Okay, well, Yolanda's going to be around, so if you have questions, you can surely catch her at the end of this meeting. Okay, the next presenter up is Pedro Rodriguez from CNAG, so I'll let him talk about what he's been doing in a long way away from here.
Okay, thank you very much. I'm very happy to be here today, and I'm grateful for the opportunity to share my experience about working as a neurologist in Sub-Saharan Africa, working with rare disease patients. So I will start by saying, is it delayed? Okay, sorry. So I will start by saying that over the last decades, progress in genomics and other advanced diagnostic pipelines has helped us to get a good understanding about the clinical genetic and epidemiological landscape of neurogenetic disorders in places like Europe. But in other places of the world, like in Sub-Saharan Africa, there is a profound knowledge gap. All these conditions hasn't been studied, and currently we lack all information about the incidence and prevalence, where the most common diseases, genes, or mutations. And this is concerning because the African population is projected to double in the next 50 years. And my worry is whether the knowledge and genetic associations that we've done in European populations are readily portable to African populations. And why am I saying this? I'm saying this because Africa is the most genetically diverse continent. Modern humans were originated in Africa around 200,000 years ago, and then they stayed in the continent for a long time until what science tells us, that a very small fraction of these modern humans migrated out of Africa, bringing with them a very small gene pool from all the genetic diversity that was available in the continent, and then subsequently they populated all the different continents in successive stages. And that's why there is this common agreement that genetic diversity decreases with distance from Africa. So taking in consideration all this different genetic background from the African populations, my question is whether these genetic associations can we transfer into people African genetic ancestry, and also whether they would be at risk of diagnostic tests being less accurate, or for instance, like novel therapies not being inclusive. And that's why I think that it's important to generate knowledge from inside Africa. I know there is a big international effort, especially now in the US, to diversify genetics, but due to historical reasons, we know that African American genomes don't capture all the genetic diversity that is available in the African continent. Even though we know that there is still a big gap in terms of representation of African individuals in genetic databases, and databases like Nomad, or this is a very nice database from Oxford where we look at diversity for GWAS data. It is still very low. So today I will be talking about CNAG. CNAG is a collaboration between CNAG and the University of Dakar. It's part of a call for global action for rare diseases in Africa. Basically, it's based on the recent advances in genomics that allow us to make a precise genetic diagnosis based on a DNA sample and phenotypic information that's being collected remotely. In particular, we can use ChipApp, which is the genome phenome analysis platform hosted at CNAG, to diagnose our patients while we do the consultation in Senegal. So this is extremely useful. And just share with you some of the videos so you get an idea. This is what we've done so far. We've established a rare disease clinic in this hospital, which is the University Hospital in Dakar, and we do this clinic like once to twice a week. In collaboration with all my colleagues, neurologists and pediatricians in Dakar. And we also do outreach clinics in the regions, because one of the main issues with families having rare genetic diseases is transportation, which is very expensive in Senegal. So far we've recruited more than 1,200 participants for nearly 500 families. As you can see here, most of the participants are children, because the mean age of the population is very low, around 18 years old. And also there is quite a lot of recessive inheritance, because there is a high consanguinity background in the population. So this cohort of patients, we've characterized them very well, both phenotypically and also genetically, with a progressive genetic testing approach. And now I will tell you about the different objectives of the project. The first one is to increase the proportion of patients with a precise genetic diagnosis and use that genetic diagnosis to improve care management plans. Using, we cannot use like fancy medication, but medication that is widely available and is cheap, and also do genetic counseling. And I have to say that we've managed to genetically diagnose more than 50% of the cohort. The diagnosis yield is greater in the neuromuscular than in the neurogenetic diseases, because the phenotypes are more complex and we still don't know them very well. And we estimate that we've made high impact interventions in 10% of patients, moderate impact in 32%, and low impact in nearly 60% of patients. So this is just an example of how important is to a precise genetic diagnosis. This is a child that I saw in the clinic. As you can see, she has drop attacks or atonic seizures, so she lost this muscle tone all the time, and this has affected already her cognition. She's been diagnosed with epileptic encephalopathy, which is a non-specific diagnosis. There is a family history of similar children affected in the family, and one passed away. And thanks to genetic testing, we can now say that she's suffering from cerebral creatine deficiency syndrome, and that we can treat this disease with a powder, a creatine powder, which is available in supermarkets or even in like these sort of like food stores, making a high impact intervention. And like this, I've got multiple examples that I can share with you. The other objective of the project is to understand what's the clinical landscape of these diseases in this particular region of the world, where are the most common diseases, what's the genetic architecture. And over the last 3 years, we've been characterizing especially the neuromuscular disorders, which are my main interest, with the different genes, different mutations. And one of the things that really stood out from this study is that some of the most common diseases that actually we see in the neuromuscular clinic here in Spain, or when I was doing clinics in the UK, like myotonic dystrophy, FSHD, CMT1A, genetic diseases that are common in European populations with a high prevalence, as you can see here. We don't see them in this population. I'm particularly interested in this disease, CMT1A. CMT stands for Charcot-Marie-Tooth disease type 1A. As you can see, it's a genetic neuropathy that causes like muscle atrophy and distal weakness in the legs and the hands. And CMT1A makes 50% of neuropathy cohorts in Europe, but in our cohort in Senegal, we haven't been able to identify this disease. It was also quite interesting to read that colleagues in South Africa, they also made the same observation. And you may think that this is related to a scarcity of neurologists, or maybe, I don't know, patients with CMT1A don't show up in these hospitals in Africa, but I do think that populations, they have different incidence and prevalence of genetic diseases, that this is related to the environment, to the culture, but also to their different demographic and genetic history. And that's what Alexandra, that was a master student at the UB, started this as her master project 2 years ago with Oscar Lau from Population Genomic Algorithm and me as supervisors. And she got a grant from IFN Telethon. And she's been continuing this project from the last year, from this year, and I'm going to share some of her results with you. So CMT1A is a disease that occurs recurrently in the population, de novo. It's caused by a duplication of this gene PMP22, which encodes for peripheral myelin protein. And it's interesting that flanking PMP22, there are like some repetitive regions that we call CMT1A reps that are 98% homologous. So during meiosis, if there is a misalignment of this CMT1A reps, they result in the duplication of PMP22 mediated by non-allelic homologous recombination. One of the interesting things is that the recombination doesn't take sort of place equally around the rep. By contrast, there are some hotspot zones, and we are particularly interested in hotspot zone 1, where most of the recombination events take place, and that's because here there is a DNA binding motif for a protein called PRDM9, which is involved in binding DNA at hotspots and making double-strand breaks and recombination. So basically, our study has been focused on studying the genetic diversity available across population in this hotspot zone 1. So we've been using population genomic data sets like the 1000 genomes, HGDP, and other genomic data sets available, and also recombination maps based on linkage disequilibrium, but also demographic history. And here you can see that the African populations, they have lower recombination rates compared to other European populations. And also the interesting thing, when we look at the haplotype in our hotspot zone 1, we can see that the African populations, they have distinct haplotypes in both the proximal and distal reps, and obviously a greater haplotype diversity compared to the other populations. But what is more interesting is that the haplotype that is most common in European population is very rare in the African populations. We've also tried to study how genetic diversity impacts in this particular hotspot zone 1 by looking at intrapopulation genomic diversity in the different populations. And here is highlighted the hotspot zone 1. And as you can see here, the African populations, they exhibit a peak of genetic diversity, this hotspot zone 1, while the European population, they exhibit a rather flat pattern. I will zoom in for you. And this is also interesting because when we look at the genetic distance, we try to understand, using all these large genomic data sets, how these hotspot zone 1s differ between the reps. we can see that there is a reduced similarity between CMT1A reps in the African population, which is significantly reduced compared to the world population. And this is of interest because the degree of homology between the reps is related to the chances of recombination. We've also been networking a lot, and I'm now collaborating with Janine Heckman of the University of Cape Town and Michelle Cavalsun, which is a clinician in Rochester but also works in Zambia, that have also made confirmed the same observation that this disease is rare in African individuals. And now we are collaborating also with David Herman at the University of Rochester, who's leading the Neuropathy Consortium in the US. It's quite important because he's got lots of data from African Americans. So he's confirmed that CMT1A is very rare in African American, but there are like 2 families apparently with this disease. We don't know whether they are fully Africans or admixed, but we'd like to probably do genetic testing in the future for these families. And anyway, I think that these results suggest that there is a population scale genetic variation that is protected against non-allelic homologous recombination events causing CMT1A. Sorry. I think there was something with my slides. Yeah, and now basically we are interested in doing different things, but what we want to do is to sequence patients with CMT1A, and we will be studying in detail using long-read sequencing the variation in this homologous region that are difficult to study with short-read sequencing. And also we are interested in, and as I said, looking at ancestry, chromosome painting, especially in the African Americans individuals, as sometimes you can have like alleles of different ancestry within the same chromosome that can, in our opinion, influence the chances of having this disease. Also very interested in a disease called FSHD, which stands from facioscapulohumeral muscular dystrophy, which is very common in Europe, 1 in 80,000 people. And actually, when I was doing clinics in the UK, we had a lot of FSHD, and when I started the clinic in Senegal, I was wondering, where are the FSHD patients here? I reached out to the literature, and I found this article from Ravi Tanwil from Mayo Clinic, also making the point that in the FSHD registry in the US, there were no participants from African ancestry, despite, 13% of the population being African. So that's a quite big deviation from the general population. And also there is recently some data from the FSHD registry in the UK where only 1 individual self-reported as being African. So I thought this is rare for African people until one finally patient came to my clinic. And then, how can I play the videos? Okay, whatever. Anyway, so until one patient came to the clinic with features suggested of FSHD, and thanks to collaborators in the Netherlands, Richard Lermer, we managed to confirm the diagnosis. And through lots of networking and collaboration, we've managed to diagnose a total of 9 individuals with FSHD and African genetic ancestry. FSHD is a very complex disease that takes place in this polymorphic D4C4 array in the subtelomeric region of chromosome 4. As you can see, normal individuals, they have from 8 to 10 to 100 units of this array, which is hypermethylated. In individuals with FSHD1, there is a contraction of this array ranging from 1 to 10 units that results in hypomethylation of the subtelomeric region and activates a gene called DAX4. So that is repressed, and that's causing the pathogenic mechanism. There is also another type called FSHD2, where the contraction of the D4C4 array, it's 8 to 20 units, but then there is another mutation in a gene that it's involved in methylation. So the interesting thing, if you look at the distribution of FSHD1 alleles in the European population, you can see that most patients stands around this zone. But what we've identified so far in the very small number of African patients is that we haven't identified anyone with 7, 8, 9, or 10 repeats. So it seems that patients with African genetic ancestry and FSHD has shorter FSHD1 alleles, and this raises like several questions, like are 7 to 10 unit alleles less pathogenic in non-European genetic background, or also do methylation levels differ across population, on how can this influence disease susceptibility? But also it poses another interesting question. Do we need to take genetic ancestry into account when we do genetic testing? Because obviously if we have someone with muscle weakness that has 9 repeats in the array, I think probably if it's European, it may be pathogenic, but maybe if that person has an African genetic background, it may be not. So that poses like questions that we may need to reflect in the future. I'm also interested in myotonic dystrophy. As I said, very common disease in European populations. It's been reported only once in African populations. Myotonic dystrophy arises from a triplet expansion of CTG repeats that actually is unstable in the germline. So that means that the next sort of generations, there is an increase in the CTH number of repeats, and the higher the number of repeats, the earlier is the disease onset. So in this study that took place in the US, in New York, they did neonatal genetic screening for myotonic dystrophy in 50,000 births. And they found no cases in children of African genetic ancestry, while the African population in New York is nearly 20%. So why is that? I mean, that's the question. But the interesting thing is that when we look at the healthy population using population genomic data sets, and we look at the distribution of CTG repeats in this particular locus, we can see that the African populations, they have a bimodal distribution, but the European populations, they have a trimodal distribution. So within the healthy population, there is a very small group of individuals. We have higher CTG repeats, and the idea is that this is a sort of disease reservoir that will perpetuate the disease within the population. But the interesting thing is that if you take another triplet expansion disorder, for instance, GPH3, this causes a disease, it's called Huntington disease type 2, and is a disease that is only been reported in Africans, but not in Europeans. We can see the other scenario. So here the Africans, they have a disease reservoir for this triplet expansion disorder, but the Europeans, they don't have it. So, I mean, my question is like, why some tandem repeats expand in some populations, but not in others? And what can we understand about DNA instability by looking at population? And also if we can use populations genomic studies to identify clinically relevant tandem repeats and novel repeat expansion diseases. Obviously, not everything is different in Africa, so this is just a summarize of the limb-girdle muscular dystrophies. We have a cohort of around 40 families, and the genotypes are very similar to what has been described so far in European cohorts. And also Duchenne muscular dystrophy is also similar to European populations, with clusters of deletions and duplications in the same hotspot that has been already described in this population. but this raised, another vision. Now we have diagnosed lots of patients with Duchenne muscular dystrophy, and I'm hopeful that in the near future we could do clinical trials in Africa. Why not? At the moment, there is only less than 1% of the trials that they are done in the continent. Interestingly, we haven't found any founder effect. When I first started this project, I thought there would be a founder effect for every different ethnic group, but we haven't found any. I wonder whether this is related to the increased genetic diversity, or whether many of these founder effects that we see in our European populations, they sort of came up within this process of populating the world. But it's just an observation that I wanted to share with you. The other objective of the project is gene discovery. Obviously, working with underrepresented populations that has never been studied is a great opportunity to identify novel disease genes, pathways, and genetic variants. And related to variants, I want to highlight that doing the clinical interpretation of exome and genome data in African patients is quite challenging, because we don't have population background data to compare, and we have a significantly higher number of variants of significant unknown significance. They are difficult to interpret, and often we need to rely on functional studies, which are very time consuming and difficult to do. But in any case, we are building a gene discovery cohort. The idea is to do patient-based gene discoveries, profit from advances in bioinformatics, omics technologies, modeling to really understand disease phenotypes, phenotypic variability, disease progression, and respond to therapies. And we've got a number of candidate genes that is increasing all the time, and I just want to share with you some of the genes that we are working. The first one is called MET20. I met this family already 2 years ago. 3 children affected with a neurodevelopmental disorder. It's a complex neurogenetic disease with motor delay, absent speech, intellectual disability, and also some walking problems in the form of spastic paraplegia. When I conducted the whole genome sequencing, I have to be honest, I totally missed this variant because it's a variant of unknown significance, VUS, and the predictors we rely on to assess whether the variant is significant or not, they still fail a lot. So it's not a very sexy variant, I would say. So I had to conduct extended trio analysis to understand that from the 13 variants that segregated within the pedigree, this was the most interesting. And actually, MET20 codes one of the subunits of mediator complex, which is a multiprotein complex which is involved in the regulation of RNA polymerase II transcription, and also works as a transcriptional coactivator. When we were doing the modeling of this massive complex, thanks to Professor Asturias in the US, basically what we found that this very tiny missense change from leucine to phenylalanine was in the interface of MET20 with a neighboring subunit, and that probably this mutation or this variant was introducing a very bulky aromatic ring into this pocket. So there was a rationale, or could be a structural rationale, that could help us to prioritize this variant. And then thanks to GeneMatcher and lots of networking, we identify a number of families harboring this variants in MET20, including one in the US with the same very variant, and also African genetic ancestry. And obviously sharing the same phenotype. So in collaboration with colleagues in France, in Strasbourg, who are doing all the functional work, we can now say that MET20 variants, they disturb mediator integrity. So here you've got the different pull-down assays where they basically look at the interaction of MET20 with the different MET subunits, 17, 20, 18, and we can see here that there is our mutation impairs the interaction with different subunits and also with RNA polymerase II. Still, we have lots of, this is another plot showing that reduced transcriptional activity. We still have lots of questions, like why the phenotype is mainly neurological if this mediator is expressed ubiquitously. This is just to finish another family with complex neuropathy. I don't think I have time to get into details, but here we found a variant in a novel gene called COX-18. And COX-18, as you can see here, it's a sort of translocate COX-2 into the inner mitochondrial membrane, and COX-2 is one of the subunits of the complex 4, mitochondrial complex 4. So this is another new gene that we are identifying. And basically, I just want to finish with the last objectives of the project, which is build capacity, train in neurogenetics, genomic medicine, and also advocacy, raise awareness about rare diseases and tackle stigmatization. This is some of the training that we've done with the different colleagues in pediatricians and neurologists on neuromuscular disorders. And I work very closely with the Patient Association in Dakar, and we celebrate for the first time this Rare Disease Day. And also, this is the Duchenne Awareness Day in 2022 and 2024. So I will just finish by saying that human populations exhibit differences in the prevalence of common and rare genetic diseases, and that they can result from different, environmental triggers, cultural, demographic, and genetic histories of the populations. I think we no longer use the concept race, because I think it's not well received. But I think we are now moving into genetic ancestry, and I think we will need to take this in consideration for precision medicine in the future. And finally, saying that there is a diversity gap in human genetic studies, and if anyone is interested in working in Africa with me, so please just get in touch. Thank you.
So thanks ever so much. That ran a little bit over. So I will ask everyone who wants to ask Pedro questions to ask him afterwards when we're done here. The next presentation is by Oscar Bolaños from Illumina, who's going to explain to us what Illumina does. Wait, here's oh, there's a lot more slides you have here. Okay, good. I think I'm on your right slide.
Yeah, perfect. Thank you. Thank you. Hi, good day, everyone. So it's a great pleasure to be here with here in CNAG with this symposium, surrounded by known faces, great colleagues, and bright minds in this genomic medicine. So today we'll try to give a flavor of Illumina roadmap for 2025. For logical reason, we'll not discuss all the details, but at least to get a glimpse of the new technologies we are in process of development. So just to start, just to summarize all the Illumina, let me check. To explain the Illumina roadmap is a bit challenging because there are so many stuff ongoing, planning and so on. So it's like, it feels like me with, so when I try to explain my daughter what is the Harry Potter book series, no? So I think we'll try to provide some insight of this new development. Just to, I will look ahead for 2025, Illumina continue to define some frameworks and a strategic direction for genomic advancement, ensuring they unlock the full potential of NGS technologies. No? So the first is just to hit a flavor of, we are in touch with in getting feedback from almost 9,500 customers all over the world. And the capacity of the generation of genome is close to 5 million genome for all our customers. So the next almost 60-70% of our workforce, Illumina employees, are focused and fully dedicated with R&D. And in addition, with our panel or IP patent pool, we are generated another extra 100 innovation this year. So today we'll like to give a glimpse of two technologies, new technologies. One is Constellation, and another is 5B. So, but before that, we'll try to recap the 2024 year. As you know, we are developed the new equipments, new technologies, new capabilities, improve the accuracy of the new sequencer. We launched to the market a new sequencer this year. We improve the accuracy. We create some faster, more flexibility flow cells in the NOR sequencing part. And also the expansion. The expansion into multi-omics is a multimodal, spatial, and natural next step to Illumina to gain increase in size of genomics, no just stay in genomics, or to expand with proteomics, transcriptomics, metylomics, spatial, multimodal data to better comprehension of some disease and translational research. Another recap for 2024, as you know, Dragon has been a benchmark in some FDA precision challenges. And also we complement these tools with AI, deep learning in different steps of the process, secondary and tertiary analysis. So it's another advancement we have developed in 2024 for the next year. For the next year, Constellation. What is Constellation? Constellation is a breakthrough technology encompassing enhanced operational efficiency and the ability to get deeper insight from genomic data. So it's an unprecedented simplicity of the workflow. So in basically, we eliminate the library prep. So the unfragmented DNA is charged in a buffer in the flow cell, directly to the flow cell. So no specific extraction method for high molecular weight for long reads and so on. So just a normal regular DNA extraction. So the DNA is charged into this flow cell and is getting some linearity. We'll see later how the DNA is spread in the flow cell and how we extract the data. So at the end is just 10 minutes workflow. We'll launch a new flow cell for NovaSeq. So this new flow cell will, this is a new nanowell where fermentation will be done in the flow cell, and we'll extract the two different data from the flow cell. This is a short read sequencing and also linearity image of the DNA in the flow cells to create more comprehensive genome data with the challenge that we are facing with short reads. This is a little bit the overview of the image. So the linear DNA will be spread in the flow cell, and we create a spatial image, so the DNA on the flow cell. So the neighboring nanowell will provide data in parallel with short read segmentation common sequencing in our platform. And we'll improve the mapping, of course, the mapping and the coverage of genome. And also we'll be able to create some longer haplotypes. Longer haplotypes close to, as you can see here in the image, with 70K kilobases for fission, improved fission. Remember, what is the common extraction method? There is no needed time-consuming extraction to get a high molecular weight for long reads. So in this case, it's a common extraction method. And also, I will share with you some images of how we'll pick up some structural variants. In this case, in our insertions, we are providing images from our laboratories that are validating our technology. We create some analytical data will be available at launch. We expect mid of next year. And another value of this technology, of course, is the cumbersome challenge of the short-reach. You're increasing the accuracy and analytical performance for phasing, also for the structural variants, pseudogenes, etc. Okay, jumping now with the 5-base genome is a methylation. As you know, the methylation is the current protocol is a sodium bisulfite treatment is needed, converting unmethylated cytosines to uracil while leaving unmethylated cytosine unchanged. So now it's a process that is quite difficult to map because at the end we are altering the DNA. So difficult to map is not automatizable. Sometimes it's very challenging process, and also the pipeline for analysis of methylation is there is an open source, but we'll try to implement all the workflow in the same pipeline in Dragon. So for the 5-base genome, we retain the integrity of the DNA. So we don't modify the cytosines, methylate cytosines. So at the end, just with a small modification of methylate cytosines for better mapping, retain integrity. New algorithms provide the annotation data for both, for variant calling and also for methylation. To summarize, it's a single library prep to get inside of the common 4-base for variants, adding methylation. It's a single step added to the complete workflow. It lasts just 6 hours. And it's compatible not just for whole genome sequencing, it's just also for enrichment panels, exon, and other applications. That is just adding this step during the process. Also, we are in the process to validate, to create some script implemented in the Hamilton in Tecan to end-to-end workflow and less hands-on time. It will be compatible with the current sequencer in the market. No extra or new equipment or new technology to implement the methylation at the same time. So this technology provides some benefits. New technologies provide richer insights with minimal coverage bias and utilize novel algorithms integrating methylation and genetic variation into one dataset annotation in a single run. So it's not needed a parallel analysis of methylated or variant calling for. So all of these solutions will be implemented in Dragon. In Dragon germline, in somatic, for, as you know, most of the methylated cytosines are, this is related to some specific rare disease, Fragile X. So Parkinson, this morning was mentioned that autism disorder, also oncosuppressor and so on. So all of these algorithms will be implemented in Dragon and also in or tertiary analysis for the straightforward analysis. So I think it's all from my side. There's more to come. Just today we would like to share just a couple of innovations for next year. There is another research in the roadmap to plan to release next year, but I think it's enough. Thank you very much. If you have any question. Any question? No, it's okay.
Do we have questions? Yes, back at the back, and the microphone is up the front, so it'll take a second until it gets there.
Thank you for a very interesting talk. Just to confirm, so with the Constellation technology, we won't need to do the library preparation, just to extract the DNA, the nucleic acid, and put into the sequencer. Is that correct?
Exactly. The INTA-DNA is no library prep required. It's the INTA-DNA from regular extraction method. So you charge the direct DNA, of course, it's diluted in a flow in a buffer, straight to the flow cell, and this flow cell spread the inter-DNA, the linear DNA is spread in the different nanowells, and we get to image the sequencing regular common short reads, and also the fluorescence of DNA as distributed in the different nanowells in the flow cell.
And the follow-up questions could be a bit provocative, but for how long do you plan to keep the normal library preparation that we usually do now?
Yeah, it's a good question. After destruction, you mean? Or in the flow cell?
Yeah, so in the future, I understand there will be the products with this Constellation technology, but do you plan to maintain the previous technology for the library prep that we currently have?
Yeah, the DNA stability after destruction is a common, let's say, stability. So you can keep it. In the flow cells, the stability is 6 hours. I don't know if you answered that question.
My question was more about for how long you plan to maintain the current library preparation protocols to use with the current machines.
Yeah, but
you don't plan to abandon the current technology, let's say, for library preparation.
Yeah, library preparation is not needed here. I don't know if you got the...
Okay, thank you. We can talk afterwards. Thank you.
Yeah.
Okay, so thank you very much. So it's my pleasure to introduce the next speaker, who is Ángel Caracedo, who is the leader of the Impact Genomica project. So now we've heard about impact projects quite a bit. This is one of the important projects in Impact.
Good afternoon. Hello, everyone. Okay, here it is. Well, thank you so much, Ivo, for the invitation. A pleasure for me to be here with all of us. I'm going to talk about about translation and clinical practice. I know that most of you are involved in research, but I am moving slightly to show you the reality of genetic diagnosis and to talk about this impact project. You know that rare disease is a concept of frequency. We call a rare disease when the frequency is with one patient in less than 2,000. It changes in different parts of the world, but that's the definition we have in Europe. There are rare diseases, but in total there are a lot because they represent more or less 8% of the total of disease. That means that around 3 million people are suffering rare diseases in Spain. And most of them are genetics, something like 85%. Because they are rare, they were sometimes abandoned at some extent in the past. That's why in 2010 we founded the IRDIC Consortium, the International Rare Disease Research Consortium, in this meeting in Reykjavik. Here I am when I was young, and the question is that the idea was to foster international cooperation in research in rare diseases, first between Europe and the States, and then there are now many advanced countries involved in the initiative as well. Every 5 or 7 years, we establish some goals in the initiative. In this moment, the goal for diagnosis in 2027 is that all patients coming to medical attention with a suspected case, suspected rare disease, will be diagnosed within 1 year if the disorder is known in the medical literature and all current, I can read from here, and all currently diagnosed individuals will enter in a globally connected and coordinated diagnosis and research pipeline. But the reality is that in Spain, we are very far away from this reality. We have an average diagnosis time, according to FEDER, of 5.5 years. Perhaps it's even worse, because there are many, many people undiagnosed so far. And the problem is it's not a problem of sequencers or machines, it's a structural problem in Spain. Probably most of them related to the organization of genetics, clinical genetics in Spain. There are very different organizations in different autonomous communities. In some of them, they are quite well implemented. In other ones are almost not implemented almost at all. Genetic counseling is a problem in most of the country, and especially education is a problem. There is no subject matters of clinical genetics in most of the faculties of medicine in Spain. There is no training for professionals in different specialties in Spain. So that's the real situation so far. Obviously, we have also some technical challenges. The most important from the point of view of the delay in diagnosis is the bottleneck from prioritized variants to the report. When we compare, for instance, an exome with a patient with a reference exome, we have a list of prioritized variants, and we have to correlate the genotype and the phenotype. This is very time-consuming, and the main reason for this is that the cases we receive, they don't have very, very clear established human phenotype ontologies or a clear phenotypic definition. So we have to go into the clinical records to read everything and to extract ontologies of phenotypes to make the diagnosis. In addition, we have a lot of BUS, variants of uncertain significance, needed of functional validation, and there is still a lot of missing variation in many groups of diseases. The good news is that a few months ago, the Ministry of Health in Spain, the Ministerio de Sanidad, published a new portfolio of services of genetics and genomics mandatory for the whole Spanish health system. This is a very good news because and they put a lot of money in the different autonomous communities to go ahead with this portfolio of services. This is a good news in terms of equity, because the autonomous communities, they don't have implemented well this type of services. Now they have to do it, and they have money as well to do it. We have a catalog for all known genetic diseases, also for cancer, oncology and oncohematology, and also for pharmacogenomics. The second good news is a very recent news. It's a couple of days ago that the specialty of genetics, of clinical genetics, was approved in Spain. Finally, it was the only European country without this clinical specialty so far, and that's an extremely good news. We were fighting for having a specialty for so many years ago, and we finally got it. There are going to be two specialties, one for medical genetics and another one for medical lab genetics. Still a lot of missing variation. The good news is that we have lots of post-zygotic mutations, especially for neurodevelopment disorders, complex rearrangements, regulatory problems in regulatory regions, non-coding DNA, deep intronic variants, new genes. The good news is that we have now new technologies, long reads, and many other new technologies that we can try to afford this missing variation. And in this context, you know that Spain received some next generation funds from Europe, and they launched here in Spain. These recovery plans are called PERTES, and in Spain the Ministry of Health decided to invest quite a lot of money in a PERTE called Salud de Vanguardia, Advanced Health, or something like that. A part of this PERTE was devoted to personalized medicine, and with lots of projects. Probably many of you are benefiting of this type of projects for research. And for this personalized medicine part, they created a and a genomic infrastructure called IMPACT. IMPACT has three pillars. One is a cohort of 200,000 people. Second, a data program that is coordinated by Alfonso Valencia from the BSc. And then the Impact Genomics program that I'm going now to talk about. The idea is they are not research programs, they are implementation projects. The idea of the Impact Genomic project was to produce data, high quality genomic data, to be shared in the European space, and at the same time to create this infrastructure to support all the research project, translational projects in personalized medicine, and at the same time to promote diagnosis for undiagnosed diseases, with the idea of with 2 or 3 conditions. First, we couldn't interfere with the national portfolio of services, so that they reach anti-exome sequencing. So the idea was to do genome sequencing, to contribute to the data to the 1 million genome project, then to do this in terms of equality, and we defined 3 use cases: one for rare diseases, another one for cancer, and another one for pharmacogenomics and population genomics. We established a network of high-capacity genomic analysis centers coordinated by CNAG, that is Navarra Biomed, CNAG obviously, and the Foundation of Medicinas Genómica in Santiago. We coordinated everything, all the standards, LIMS, quality controls, procedures, to work as a single unity with extremely high quality standards. The idea was for rare diseases to try to analyze people with undiagnosed diseases after exome sequencing. And for this, we established a network of 110 hospitals covering the whole country with coordinators in the different autonomous communities. We have many people involved, lots of clinical case managers, and also we involved the Patients Association for Rare Diseases in Spain is called FEDER. For us, it was extremely important to work with high ethical and legal standards. I was very worried because some European projects in genomics, they can't share the data they produce because the lack of ethical and legal standards. So we work in this part a lot. We have Carmen Ayuso coordinating this part together with Pilar Nicolás from the Basque Country. We produce models of valid informed consents for diagnosis, also for minors, disabled people, and diseased people, and it was mandatory to participate in the project to have informed consents for secondary use of data. All these consents, of course, they were approved by ethical committees, and then we also signed MTAs and DTAs agreements with the 3 sequencing centers and most of the hospitals and the autonomous communities. We defined 2 circuits, one for diagnosis. We required negative exome sequencing to enter into the program. And then the clinical centers were collecting the samples and the clinical information. The clinical information was handled in a clinical database, and then the samples went to the sequencing centers. We produced the reports. The reports went to the clinical case managers, and they produced the final diagnosis report. Then we have a second circuit for the secondary use of data, with the idea to have this in the future, all this data in a federated EGA in Europe. We have some transversal groups because we standardize all the diagnosis procedure, the laboratory reports, the bioinformatic analysis tools, the clinical reports, and the data sharing procedures. It was extremely important, the clinical phenotyping platform that is established in the CIBER. We have a good basis for this because we have a platform in the CIBER that is a network in Spain for research in rare diseases called ENOD, that it was the basis for this platform. And it's important that all this clinical information is not linked to the genomic information. And I'm going to talk about phenotype ontologies later on. We defined a common tool after a competition for prioritization and analysis of variants. After we were checking different tools, available tools, and finally we decided the GPAP from CNAG. It's a very good tool. They need some fine tuning at the beginning. We work together with them to improve the tool, but the tool could be implemented in the local nodes, and the good thing is that we can work all together exactly with the same common standards. Finally, we integrated or interconnected the phenotyping and analysis platforms in the infrastructure. With the idea to have all this in the EGA node in Spain, and what we do was for rare diseases to analyze 2,000 patients, most of them in 1,500 in Jingleton, 500 in trios with whole genome sequencing. We do the same for inherited cancer. And with 172 patients, with also in trios. Also, we analyze the tumor with RNA-seq and whole exome sequencing in the case of tumors. We decided also to implement a pilot project in somatic mutations, analyzing primary cancer of unknown origin. That is a problem because it's not in the portfolio of services of the health system, and the patients could benefit of this type of advanced diagnosis. I'm not going to talk about pharmacogenomics and population genomics because I don't have time today. Extremely important was the implementation of expert committees. We involved more than 400 experts covering more than 1,500 genes and diseases and in system categories. Extremely important for the future because the future is data mining of the clinical records to extrapolate to HPOs, ontologies, but they were not defined in Spanish. We translate into Spanish 17,000 labels, terms and subterms of all HPO concepts. And we produce and we give training workshops to clinicians, to many people. It was an extremely good phenotypic definition in the cases we have, with an average of more than 11 HPOs per case. That's extremely high. We finished all the equipment and all the clinical information. We have all disease categories. Most of them were neurological and neurodevelopment, 70% more or less. The people, they have an average, the patients, they have an average diagnosis delay of almost 11 years, and with an average of 3 previous genomic tests, negative all of them. At the moment, we are still analyzing the cases. We have analyzed 27% of the cases, but not completely. We only do SNP SNVs in dolls in the 100% of the cases, of these 27% of the cases. We are obtaining a diagnosis rate so far of 21%. That means when we finish the analysis of CMVs and STRs and splicing variants, probably we are calculating a diagnosis rate of 30%. imagine that the normal diagnosis rate in the lab for a rare disease is 30%. So we are duplicating the diagnosis rate. And this is not only because of the genome analysis and the tools, it's because of the HPOs, the definition of the phenotype. I'm not sure the contribution of each of the two different things. This is the situation so far. Genome sequencing has been completed. The GPAP is ready to be used for all these things. We are doing functional validation in many other cases. In some cases, we got the result, the diagnosis using final validation. I'm not going to talk about cancer because it's more or less the same. Inherited cancer is more or less the same type of circuits, only that we have to include tumor boards for patients' inclusion and for the diagnosis. And but it was more or less the same thing. We have most of the cases we have were breast cancer, followed by colorectal cancer and endocrinological tumors, and we obtain a diagnosis rate. We have finished all the analysis so far, and we obtain a diagnosis rate of 15%. That is quite a lot. The average diagnosis rate in cancer in clinical practice is 25% in inherited cancer. In primary cancer of unknown origin, the important thing is that we have set up standards for somatic mutation. It's extremely difficult to get common standards for somatic mutations, and if different labs are doing different things, it's extremely difficult to compare results. We have produced common standards, and we have working a lot in the labs because we are doing exome sequencing, not panels. And we were able to produce results in an average of 19 days. That is pretty good. And the extremely good news is that we obtain results in almost all the cases. And in 27% of the cases, they have actionable results. That means the patients have benefit of therapies because of the analysis we have done. We have involved the patients from the very beginning, involving the patients' associations and disseminating the information to promote equality. And finally, we have been granted with an extension of the impact program for the next 2 years. We are very happy with this. It's more or less the same thing because the call was for rare diseases. We have excluded pharmacogenomics in this new impact. We have included a working package of rare tumors to follow with the effort in inherited cancer. And we have implemented 2 new working groups, working packages, one for functional genomics. This is extremely important because there are lots of dispersed efforts in the country. We need good pipelines of functional genomics. And finally, the idea of the program is the sustainability in the near future. So we need to translate this to the national health system, and of course promoting citizen participation. We are in this new impact. We are going to try new technologies. We are going to do RNA-seq, especially long reads, methylation, genomic optical mapping, to check if we are able to follow, to trying to decipher all this missing variation. And that's all. It has been a huge effort for so many people. There are more than 400 direct collaboration, but more than 1,000 people all over Spain involved in the project. If we include the experts that are participating in the project, we have meetings every week for everything. For clinical diagnosis, all the cases are discussed in meetings. So this has not been for me the most important, not the most funded project in my life, of course, but the most important by far, because it is for me it's really, and I have to thank all the people involved in the project because it has been an effort from the very beginning, working all together. Imagine in a country where the regional differences are impressive, to be able to help people with so many new diagnoses, to obtain high quality data, to be shared in the European health data space, and at the same time to promote common standards for having better diagnosis in the future. And that's all from my side. Thank you so much.
Thank you, Andre.
You're very welcome. I don't have voice, I'm sorry.
Yes, you do. Questions? There's no questions. People not asking a lot of questions today.
Better for me because I have to leave.
You have to leave. Okay, good.
Well,
thanks.
Thank you.
Good. Okay, so it's my pleasure to introduce Andres Ramirez, who comes to us from 10x Genomics. And I see this works. This is really
good. Thank you very much. I work for 10x, but today I am representing our distributor, Bonsai Lab, who sponsored this symposium. So thanks to them. And I just want to share some reflections and some data in what we think at 10x is the direction we want to take into bringing genomics into healthcare provision. Where do we stand in regards to this topic? It's pretty much where we all stand probably, which is an incomplete understanding of disease leads to poor diagnostics on one hand, as we saw in the previous presentation quite well actually. But also from the development of the therapies, new therapies, having more limited repertoire of potential targets, for example, that we can address with new drugs. Ultimately, in the end, patients and families that have not many options sometimes to try to go through some complicated diseases. And in the spirit to share a little bit what can be done with our portfolio at the current state of the technology, I brought to you a little case study, if you like. This is a paper that was, this comes from a paper published last year on Nature Communications, where basically we use our toolkit, both single-cell RNA sequencing and spatial transcriptomics, to explore a little bit breast cancer biology. Why cancer? It's kind of obvious probably. It's highly heterogeneous, it's difficult to understand, and this is why spatial transcriptomics especially are so highly relevant. So we took some blocks from a tumor that were preserved in FFPE, probably the option of choice in most of the clinical settings, so these biopsies are just stored until they're used. And we try to show here how a more comprehensive view using all these tools together can actually bring more data to the table and always in order to provide sometimes better diagnostics or better understanding of what we have at hand. So this a little bit about the methodology. Basically, we received a block of a patient that was diagnosed in stage 2 breast cancer. There are some classification markers. We have, for example, the receptor of EGF-beta, receptor 2, the receptor of progesterone and estrogen. So in this case, this patient was a double positive, was diagnosed as positive for the estrogen receptor and for the receptor 2 of EGF. So what we did was we took a 25 micron curl of this tissue, and this was dissociated and was run through our Chromium platform for whole transcriptome profiling. And then 2 consecutive slides of 5 microns each were analyzed with our Visium spatial whole transcriptome solution, and then another one for Xenium in situ, which is single cell in situ transcriptional analysis. And what we found on the left panel, we have the results from Chromium single-cell data. Basically, the unsupervised clustering revealed the 17 different clusters into this tumor sample. And these clusters, basically all the typical cell types you expect to find in breast cancer, such as stromal cells, endothelial cells, and immune cells. On the right side, you have the results from Visium, which is basically the same, but adding the transcriptional, the spatial component. Interestingly enough, both Chromium and Visium were able to identify that inside the cancer cell compartment, actually we had 3 different cell types. 2 different types of ductal cancer in situ, which is considered to be a stage that's prior to invasive cancer, and a 3rd type of cell that was invasive, clearly invasive. And so taking the single cell data and using this to interpret the spatial data, we could get to this Visium spatial plot, where we can identify, I'm not gonna go through all of the data for the sake of the time, of course, but you can see that these ductal carcinoma type 2, type 1, they have different spatial distributions on this grid. And then we could also identify adipocytes that we could not see in Chromium single cell, because these cells are more fragile, tend to stick to plastic, difficult to retrieve from the sample. However, they are visible there. So the problem is that although we found the same number of clusters in both platforms, 17, there were 7 clusters on Visium that were actually a mixture of different cells. Because these transcripts are transferred into spots, and the size of these spots is much larger sometimes than the cells. So we ended up with mixtures. But to resolve this better, we have this 3rd tool, which is Xenium Spatial. So Xenium is able to localize different transcripts and to assign them more precisely into individual cells. So we have now single cell resolution on the spatial context. And this is just a representative image from this data set, highlighting a little bit some known cell types in close interaction that we can very precisely distinguish. But if we go back to our tumor and we zoom out a little bit, here, this is where things get a bit more interesting. As I mentioned, we knew from the single cell data, and we could see the same on Xenium, that there were actually a bit of diversity in the cancer cell compartment. So two different types of ductal carcinoma, one type of invasive tumor. So we asked a pathologist to take a look at the sample and to identify these ductal and these invasive cancer types. And this is what the pathologist could annotate. He could see there were differences. He could see the ductal carcinoma and the invasive cancer lesion on the tissue, but no way with amidoxolin L-Seq to distinguish this heterogeneity between these 2 different regions. If we compare with Xenium on the left, Xenium allows us to distinguish these 2 types of carcinoma, and we can see them annotated. So if we overlap one on the other one, you can see the outline of these regions in different colors, the color code for the invasive tumor in red here, and the 2 different types of ductal carcinoma, one here in pink, and the other one more in orange. It does not look so orange here, but believe me, it's orange right there. So this is something that's very powerful, because we knew that these cells had a different transcriptional profile, but now we can select a region of interest inside a tissue that has pathological significance in the eye of the specialist. And now we can ask ourselves, what else we have in this region? And actually, we could see that the cellular composition between the 2 different ductal carcinoma regions were different. They were both enriched in myofibroblasts that were absent on the invasive cancer region. But at the same time, the ductal carcinoma region 1 was enriched in some genes that belong to the invasive carcinoma, suggesting that we are seeing in real time actually the progression from ductal carcinoma to invasive on one slide. And not only that, if you remember I started describing the block we were working with. So this block, this patient was diagnosed to be a double positive. And upon closer inspection on the slides, we could find an actual region that was triple positive. So this patient was incorrectly diagnosed, basically, not because of negligence, but because there is an actual region that's very small that has expression of these 3 markers that could not be seen by the regular inspection. So if we do a little bit of a mental experiment here, and we think of a hypothetical therapy that's targeted, we go a little bit on the future maybe, and we think of maybe some carriers, some that could do specific homing maybe to ductal carcinoma type 1 or to double positive cells. We're totally going to missing this one. And maybe we're just applying a selective pressure and we're making this tumor change. And then the person has a relapse with a different type of cancer afterwards. So this is something that can be analyzed backwards now, and because Genome is targeted, we use a specific panel of transcripts for analysis. We can go back to the whole transcriptome data in Visium, and even though the spots are big, we can still overlap, and we can still analyze specifically those spots that contain these cells, and we can find genes that are specifically being regulated on this specific population. So as you see, this analysis I showed very quickly, very high level, I know, but basically highlights what can happen when you use different tools in combination. We believe a lot in synergy. We think that using tools in combination brings more data, more insights, than just sticking to what you can conclude from each one of them individually. And let's consider something. This, in particular, this population identified with Genome was done with a 313 panel. So we took our regular off-the-shelf 280 gene panel for breast cancer, and we added 33 genes on top of that, and we ended up finding this population. Now we're moving into higher plex, and now we have 5,000 plex, for example, available. So we can only imagine, and we cannot wait to see what researchers like you are going to do with these ever-changing and ever-evolving tools in the future. So having said that, this was the toolkit that we use in this trisection, not your typical dissection, of course, but this a little bit to show what can be done. And with that, I thank you for your attention.
Does anyone have a question? No. Okay, thanks ever so much. The final speaker of the day is Thomas Marquez. I can't see him. Where is he? I can't see Thomas. Let's see if we can find Thomas Marquez. Someone ran out to get him. Ah, here he is. Good. You see, you should have come in, with da 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 da. I was going. No problem. Good. Thomas Marquez.
Yeah, sorry, Ivo. Hi. Sorry, everyone. I had an emergency I had to take care, and I didn't realize it was that late. So, first of all, I would like to thank everyone in the room for many reasons. One is because this has been the longest session without a bathroom break, and I think that many of you are needed to take a break. So thank you for staying. So the second thing is that I'm the last speaker on a symposium that is about healthcare, and being the last talk is always challenging to try to keep you a bit with me. And the last thing, just in case any of you is not paying attention, my talk is going to be about primates. So if any clinician or if you guys are not interested, I will not be offended if you guys just leave the room and That's totally okay with me. I mean, it has happened before, so that's not a problem, okay? However, my challenge for today, and this is, I take that as a serious challenge, is to try to convince you from another perspective. This is going to be a very different talk of the ones that you have had before, but I will try to convince you that even from a clinical perspective, sometimes it's good to just think a little bit out of the box and go to another forms of life, in this case the primates, trying to get some answers to problems that has not been solved yet. And I think that non-human forms of life can really provide answers to specific questions. I just want to be very clear about what I will not be doing. So the talk that you will see today will not cure any patient, okay? And I want to be particularly clear about that, because me and my group and the people who work on non-human primates, we need to acknowledge that. Although, of course, in the grants we all put, this is going to be very whatever, right? But I think we need to acknowledge that when you guys, clinicians or people like CNAG that are running super important diagnostic tools, all these tools, they do not come from nowhere. They are based on some fundamental biological problems that many times for all of you is an algorithm like grep score. Okay, it's just a conservation score, right? And my mission for today is to try to explain to you one of the algorithms that it's been used and will be used. And I want you to be aware that when we are making diagnostic and we have that mutation that finally it's going to provide an answer to the genetics, to the patients, to the families, even for genetic diagnostic, the way we have detected these mutations many times requires people like me and many others who are evolutionary biologists, who are primatologists, and we try to do something that looks simple but sometimes is not that obvious, which is how to reconcile the human disease with evolutionary biology, okay? And to my knowledge, this is one of the projects where I think we are achieving something that my lab and other people has been trying to get for many times. So I'm not just sure this is the right presentation, but I will go through Lydia. I'm not sure this is the right one, but I think I will get to the end. So the project I want to present to you is this project that was a fantastic experience, and it's been a long journey and it's still going on. And essentially it's a project that's been a combination of different people, very important. Here on the left, this guy here. This is Kyle Farr. Kyle is one of the vice presidents of Illumina and is the head of artificial intelligence there at Illumina. In the middle, there is Jeff Rogers, who is at the Baylor College of Medicine, and myself. And I would like to stop just for a few seconds and acknowledge very strongly two people here in the first row, Marta and Ivo, because they have been absolutely instrumental in this project, and also Illumina Spain. I think Marta was here. I don't know if she's there. Yeah, she's there. So I know that this project has brought a lot of headaches to a lot of people, but I'm super happy that that was a reality, because as you will see, what we have achieved together, I think it's quite significant, right? So essentially, in this project, the idea was really straightforward, which is, yeah, this is not the right presentation, but it's just that I'm seeing the next slides, but don't worry, we will do something with that. So when we start this project, there was only a few dozens of primate genomes with whole genome data available, okay? And again, I'm trying to bring you to this kind of thought that integrates human biology and evolutionary biology. And as you know, humans, when particularly from a disease perspective, we try to think about ourselves like we are an isolated box, and we care case control, that disease, not disease, population. Even there was that talk that I like a lot about the African diversity. And so things like that, but it's always human as a whole, right? But for people like me, human is just only, in principle, another form of life. In this case, it's just another primate, okay? So if we can make use of the other non-human primates to illustrate about human biology, that will be a big success. And this is something that my lab has been defending for many, many, many years. And then in this project, what we wanted to do is to go particularly from the few dozens of primate genomes that were available in 2020. And the idea that Kyle, Jeff, and me had was, let's try to cover as much as possible in a really short period of time, trying to get as much primate genomes as possible. And essentially, here this is the conclusion of the project. So in less than 4 years, or it was 3 years of intense production, we went from 57 species to 233 species of primates, okay? That sounds easy, so the plot, I think it's not here, but believe me, this took a lot of coordination worldwide. I mean, there were 21 countries involved, hundreds of researchers. I know that for those of you working with clinical samples, there is a lot of bureaucracy and administrative work you have to do, and I'm very well conscious of that, but at least you have something that we don't have, which is the consent agreement. We don't have consent agreement for primates, but there is a lot of legislation for endangered species. I need to deal with CITES, I need to deal with Nagoya protocols, there are MTAs, there are MOU, all of that are things that are happening every time a single sample is moving between regions, right? And these require indeed a lot of work. So getting 233 species of primates in a single project was something remarkable, and I need to acknowledge a lot of people that was really involved into getting these samples. So this is a bit what we cover. So essentially the production data, and again, I want to thank CNAG and Illumina Spain, because 80% of what you see here was produced here in Barcelona. And essentially we generate over 800, well, we generate 700 and a few high coverage genomes, and there were 100 primate genomes already available, and most of them was done here. Something that was remarkable is that, of course, we include all the families, most of the genera are in there, and again, more than 50% of the species worldwide were covered. Among them, 30 of them were critically endangered. So for those of you who are not into that, which I assume it's the majority, there is a red list by one association, which is the IUCN, that makes a catalog of the next species that will be disappearing in the next years, right? And the last step before extinction is critically endangered, and of course that was one of the targets of this project. As you can see, the distribution is not even. We select baboons as a case scenario to do population resequencing, deep population resequencing. I'm not going to talk about that, but baboons are fascinating both for biomedical research and from evolutionary and population perspective. And then there was, in this case, 33 species with more than 5 individuals. This is important for the goal I want to illustrate. So going quite faster here, because I want to go to the disease, which is the motivation and why I'm here. But of course, after you have this data set, there are quite a few things that you can start exploring. One of them is, for the very first time, we can assess the diversity of the primates worldwide. And please bear with me in this slide and then in the disease slide, because this is what made sense of this project, and that's the foundation of the project, which is, if in this bar here, you have essentially the heterozygosity, so just the standard SNP variation between the two chromosomes. And as you can see, even in families or at territorial level, most of the primates have between 2 to 6 times more diversity than humans. Humans, as you know, the typical diversity out of Africa, it's 1 per 1,000 base pairs. In Africa, it's slightly higher, but not that much. And essentially, you can see here the variation in primates, and you can see here at the bottom the dashed red lines for African diversity and non-African diversity. So the point that you need to take in order to follow in the next slides is that humans, because we are a super recent species, you were mentioning the 200,000 years old of the out of Africa and all of that, we have very recent diversity. And remember, if we are trying to solve or contribute to the biological consequences of mutations in the human genomes, cases and controls within the same species of very low diversity, that's not what you will target in an experimental design, right? And in this case, primates can help us a lot. The other thing that we have done is a good phylogeny. This is have both taxonomic and association purposes, because if we want to do trait association, tails, brain size, diseases, it's very useful, well, it's very convenient to have a proper phylogenetic reconstruction. And this is the first time that that can be done at whole genome level. Something that has been of interest for people like me is the amount of human-specific mutations, and my group, like many others in the world, has been trying to understand the amount of amino acid and genes that contains truly derived human specifics, and the way we have been traditionally doing that is by comparing apes, which was one of the mottos in my lab, to Neanderthals, Denisovans, and humans. And now with 233 species, what we found is that almost half of the things that we thought they were human specifics, they are not. They were at least seen recurrently in 2 places in this phylogeny, meaning that, of course, that helps to reduce the number of mutations and the number of genes. Now everything is encapsulated in 80 genes that contains truly, at least with 233 species of primates, human-derived missense mutations in genes. But let me briefly talk about the consequences for you guys, or at least what I think. Again, I'm not going to cure a patient, I don't see patients, but I do hope that this algorithm will contribute for you guys to make better diagnostics. Again, one of the main challenges in biology, and we have seen that, and I think that different speakers have been talking about that, is that we don't know the functional consequences on amino acid variation. I mean, I had a graph here, but I can explain it to you because it's very easy. Out of the 70 million of potential amino acid substitutions that one can see in the human genome, we have only functional interpretation for less than 1%. 99% of them, we still don't know what it means having this amino acid change. Yes, we can of course interrogate 3D variation. Thanks, Mark. We can do a lot of things that help us to determine the functional consequences, but not in terms of how benign or how damaging that mutation can have in terms of consequences for cell biology. And this is when the primates comes into play. Essentially, in this plot, what I'm showing is one portion of the human genome, and here on the top, you have all the potential amino acid variation that has been seen in human population. Okay, so each green dot or cross here, it's an amino acid substitution. And here below, it's the clean bar pathogenic mutations. So those are the mutations that we know that when you had that amino acid, it has a case, and you guys has essentially diagnosed someone and associated this particular mutation with a particular disease. And essentially, you don't need very fancy statistics to say that there is non-overlap between this track here in the middle and the clean bar. This track in the middle is essentially whether an amino acid is seen in other primates at moderate frequency, yes or no. Essentially, what this plot shows is that when something is in clean bar, is not seen in primates. And the other way around, when something is common in primates, it's not the basis of our disease. And again, this goes into that integrative view where we as a species and as primates, we share with them physiology, cell biology, chemistry, and everything. So getting all this information and all this variation that we don't see in the human genome, but we see in our close relative, can be very informative in order to just partition the amino acid variation into benign or damaging, okay? So, and after we can do that, we can now train algorithms to learn the properties of these tracks of the genome compared to these tracks of the genome. So, as you know, artificial intelligence, a very fancy word, it's just algorithms that, based on training data sets, they learn certain patterns, and then you can apply it to other parts of the genome. The problem, or one of the problems that we had in this challenge, is that we didn't have a good set of benign mutations. Again, 99% of the amino acids, they didn't have a classification. Now, just simply overlapping the frequency in primates, we were able to classify 4 million of amino acid substitutions that before that we had no idea whether they were benign or not. And that was the training data set that we apply into a new algorithm that it's called Primate AI, which essentially now outscores all the other ranking algorithms when they are trying to define new mutations that you see in your patients, whether they are benign or damaging mutations. We propose that using the frequency in the primates, it's one of the best algorithms we have in order to provide rankings of mutations. So just to wrap up, all this was a really good amount of work where, again, hundreds of researchers were participating in that, and we were lucky enough that Science devoted a special issue in there where we published 5 papers, and then a few months later there was another one in Nature where what we did is just a global multiple sequence alignment of all these 239 species of primates and humans, and we detect the sites that are conserved, also again increasing the algorithm of conservation through evolutionary distance, trying to find those mutations that when they disrupt all these tracks of conservation, they are usually involved to disease. And that's also again now part of the algorithm. So this is not over. This is what I presented as a phase 1. This is how we call all these first 800 genomes. But now we are in a position to say that we are now closing and freezing what we call the phase 2. The phase 2 is now over 2,300 individuals from 304 species. And as you can see, compared to that, it's very challenging now to get new species into the database, but now essentially what we have is a lot of population resequencing for a lot of species, which again, people like me, we care about them. I would like to increase conservation genomics, evolutionary genomics, but I am also, of course, very happy to see that essentially they are being used to help diagnose in disease. This slide we can skip, so I'll go to the acknowledgements. This is essentially my group. I would like to acknowledge, of course, the people in Illumina. So this is Lucas Kuderna. Lucas was a postdoc in my group, and he's now working at Illumina in Kyle's team. And again, I would like to particularly, again, thanks Marta and Ivo for all the help received during this project. Thank you so much.
That's it. Okay, thank you.
No, stay here. Questions? You see your question.
Mark.
Gracias. Thomas, my question is, what you have shown us is at the level of the protein, right? Amino acid changes in the protein. So you have such a plot for all the protein coding genes in the human genome, I guess, where you compare this. But what about more recent genes that have appeared more recently in the genome? Do you have the same level of accuracy, or you really need to have genes that are old, evolutionarily speaking?
That's an excellent question. So what I present has many limitations, which we are trying to overcome. One of them is the non-coding variation. I know you're not asking for that, so I'll go to your question. And that is why that alignment, multiple alignment of all the primates to the human genome was to detect conserved regions outside genes that are preserved or are derived in the human, just to try to illustrate the conservation score and new mutations, particularly in disease, outside of genes. That was one of the caveats that we had. And the other caveat, and you're totally right, are the derived new gene structures or gene duplications. So all this project was based on one-to-one orthologous between human to lemurs, so all the primate radiation, but of course there are many gene families. We have talked about spinal atrophy and all these genes duplications, and that is not yet part of what we have done. In part because, although we like a lot Illumina technology, thank you so much, you know that copy number variation is challenging, even with read-def. So there are some people, including in our group, in collaboration with other groups, that we are doing now a map of copy number, but at the end we will have copy number, and it's very difficult to ascertain all the paralogous mutations, particularly in gene-specific duplications that we know are related to many diseases. So this is a challenge, and we are working with that, but at the technical level, in this specific case, the short read is complicating particularly the structural variation in derived scenarios like the human genome compared to the rest of the primates, right?
Yeah, thanks for this interesting talk. I was wondering if these missense mutations that are not present in the primate genomes, if you could also associate with alpha-mut or other missense predictors, and also in this way you can see for benign predictions. For alpha-mut, for instance, that is also a missense predictor tool. Also, you can predict if it's a benign or not benign, no? If you could correlate this primate missense prediction in another predictors.
That's a great question. That's something that we have been thinking about. So what we have done is a comparison of missense predictors between primate AI and others, and that's done. But I think your question is very interesting because, of course, otherwise I will not be here talking, everyone has in mind the human-derived mutations and the consequences for the human-derived mutations. But you can now play with many other interesting combinations. For instance, something that we are doing is at which extent all this variation can inform in model organisms that are being used in disease. And I'm talking about baboons, macaques, marmosets, or even mouse or beyond mouse, right? So this whole project was designed, again, trying to use all the primates to inform about one species, that is humans. But we can use the other way around, and we can use other predictors or other species just with the same algorithm and the same idea that we are putting forward here. And it works. We have preliminary data that we can use that for other organisms that are not just human, right? It's just that the most obvious way to apply this idea was to compare human with primates, right? Thanks.
Thank you. So I assume the reason why the primates doesn't have the mutation that are clean-bar in the human case is because they live in a very wild environment. So the mutants are they are going to be killed, right? So if that's correct, is not that somehow also a limitation in the sense of a mutation that is bending for not. Like, for example, why if we carry mutation that will kill a primate in their environment, but we can have the mutation until, for example, we age, and that's when that mutation becomes pathogenic.
Yeah. So your question contains different concepts, and I will try to deal with them one at a time. The very first thing is that precisely this method works because primates I mean, we have a common ancestor with all the primates pretty much 35 million years ago, and after that we have followed different evolutionary routes, right? And then, despite of these opportunities for mutations to arise and for natural selection to remove them, what we see today in their germline mutations is something that has made these animals healthy in principle. And that's the idea that when something is very common in all the primates, it's most likely to be benign in our scenario, okay? However, and now you open a second door, which is way more complicated. There are diseases that are not, you know, the standard Mendelian one gene disease, you get the mutation, you are sick. And that's more complex a scenario. And here, things like epistasis or interaction between different genes is when it comes to play. And there is a whole group or subgroup within our big group, not mine, I mean the big consortium that is looking precisely at 2 hits, 3 hits, epistasis and all of that because of what you were saying, because some of the mutations that we have as an adults, they allow you to reproduce and they are onset mutations that appear later on, right? And for that, there is another different genetic model that you need to take into account. But this model was designed to rank mutations that are in principle one hit, one gene and disease, particularly in somatic mutations. So something that appears not in your germline, but in other parts of the body, and that is the driver or potentially cancer, for instance. Right.
Thank you.
You're welcome.
Okay. Thanks so much.
Yep. Thank you.
A lot. Thanks a lot, Thomas. So I hope it. I hope it sort of starts making sense to everyone why we put Thomas' talk at the end of the day. The reason was he's a very amusing speaker. That's one, obviously. Should have stayed around just to watch that. But no, the content and mainly this bridge that goes between using this primate project into human health, which I think is really extremely important. I want to close the symposium by thanking the sponsors. You know, we have obviously received good money because we got nice food for lunch and we had nice coffee breaks and so on. So that was all paid for by Illumina, Nanopore, Bonsai Labs through and 10X, then Agilent, PacBio, and Roche. Roche themselves did not want to come and give a presentation. I think we didn't have space to give them a presentation. I don't remember. So the sponsors are really essential for us to actually make this symposium happen. The second thank I want to extend is to the speakers of the day. That is obviously much appreciated that they came and gave presentations. I think we've had a very intense day today, and I basically want to put my hands together and thank them all for their contributions. Really much appreciated.